Great.
Good morning. Welcome to the second day of the conference. I hope you all enjoyed yesterday's session. So we'll get on to with today's. We have 8 talks today, and it will follow. It will be followed by a panel discussion, just like yesterday. I just want to remind everyone again: put your questions in Q&A. If you have any problems, put it into the chat. So Candace and Jonathan can help you with technical issues. And speakers, you do not have access to Q&A box, so you have to unmute yourself and just ask questions directly. All right, we will get on to our first speaker today. Alejandro Chavez from Columbia University. He's an assistant professor in pathology and cell biology. And his title is Controlling Gene Expression with CRISPR-based Tools. Welcome.
Thank you. Thank you. Let me share my slides. Full screen. Can everyone see that?
Yes.
Awesome. All right. Great. So yeah, as mentioned, my name's Alex Chavez. I'm assistant professor at Columbia University. And I'll tell you about some of our work on building CRISPR-based epigenetic modifiers. So I'll talk about 2 technologies today with you. One is our work on building improved CRISPR activators, and another is our work on building improved CRISPR repressors. So sort of both sides of the coin, turning genes on and turning genes off. So a little bit of Cas9 basics, I'm sure everyone knows. And if you didn't know after yesterday, I'm sure you do, that CRISPR is an RNA-guided endonuclease that goes and it'll bind to DNA. And once it binds to DNA, it'll cut both strands of DNA. But what else can you do with CRISPR aside from cutting DNA? Well, to mutate the residues within the protein that are involved in that catalysis, you can make what's called dCas9 or dead Cas9. And so dead Cas9 can still bind to DNA, but it just won't cut the strands. And we sort of equate this in the lab to like a molecular couch potato. You know, it's kind of lazy, it kind of just goes to DNA and sort of sits there. And what you can do is you can then direct that little couch potato nearby to a gene's promoter. And by fusing either an effector domain to the dCas9, you can get either transcriptional activation or transcriptional repression. And so why should you care about dCas9 in particular activators? So cutting is obviously amazing and very powerful, but has a few downsides. Cuts for now are pretty much forever. They're hard to reverse. They're not 100% specific, so you might be trying to cut one gene and end up cutting 2 or 3 others. In addition, some cells just hate cuts. So cells that are p53 positive tend to not really enjoy it when you cut them. In addition, using cutting to actually turn a gene on is a little tricky because usually you need to involve some sort of promoter swapping or know some sort of repressive regulatory domain you can knock out. And so again, it makes it a little tough to get activation from just cutting alone. And where do we see activation as particularly useful? Well, I think to understand gene function, activators are amazing because you can then turn the gene on and ask what happens. Also very good for modeling things like gain of function mutations with cancer and performing programmed cellular differentiation, so turning stem cells into your cell type of interest. And so when we started this project, we certainly weren't the first people to think about dCas9 and dCas9 activators. And so what was known is that they definitely worked and they existed, but they just weren't very potent. So here I'm showing you a set of different guide RNAs against the same target gene. And what you'll see is that most of them don't really get much activation. And so usually you need several guide RNAs with the previous technology to get a good amount of activation. And even then, you're only gonna get about between 5 and 50-fold induction of your given gene of interest. And that's with 10 guide RNAs in this case. And so what would an ideal Cas9 activator look like? Really, you'd want it to work with just one guide RNA, and that's important because that lets you then do genome-wide screens where you can kind of tile every single gene in the genome and ask when upregulated, what does that do? It also allows you to get robust gene upregulation. You want to know that when you're actually using your tool, you're actually going to turn the gene on, not that it's just failing every once in a while. And so for our project, we set a goal of 100x induction for the majority of targets we were going after. And so what was our experimental setup to discover this new tool? We basically searched the literature for transcriptional activation domains. We then fused them to dCas9, and then we tested them against a nice fluorescent reporter. The way our fluorescent reporter looked is it had a TD tomato fluorescent gene, which was downstream of a minimal CMV promoter, and had a guide RNA binding site upstream of that. When dCas9 plus the activator binds to that construct, if it's able to turn on gene expression, the cells will turn red. It's very easy for us to quantify that using flow cytometry. And so how do we build this whole thing? So on the y-axis, I'm showing you the output from our fluorescent reporter assay. On the x-axis, I'm showing you everything I tried fusing to dCas9. And this is the gold standard at the time when we started the project, VP64. So you see it does certainly turn on the reporter a decent amount. And this is what we saw when we tested everything else that we were trying to fuse to dCas9. And so One thing we learned from this experiment was that most of these domains kind of stunk, which I thought was actually quite surprising because VP64 was discovered, I think, in around the 90s. And so I thought to myself, what the heck's the chance that someone discovered the ultimate activator like back then? But apparently they did a really good job because this experiment was kind of a dud. In addition, no superdomain was found, which was kind of what I was hoping for, again, thinking that VP64 can't be that good. And so at this point in the project, I got to say I was kind of depressed, thinking to myself, oh my God, this project ended before it even began. But before we totally threw in the towel, we thought to ourselves, well, how does the cell endogenously turn on genes? How does it do that magic? So it turns out that endogenously, the cell doesn't use one transcription factor to turn on a gene. It uses whole families and whole sets of them. And what happens is that those transcription factors then cooperatively recruit the RNA polymerase machinery, and that's what actually leads to nice transcriptional activation. So for example, if you have a promoter that binds two transcription factors, you're going to get kind of weak RNA polymerase recruitment and then gene transcription. If you have a promoter that binds more transcription factors, again, they're going to kind of pull on RNA polymerase a little bit harder, and you're going to get more transcription of that locus. So we had a very simple hypothesis, which was, well, why can't I just try fusing more of these domains to DCAS9? Who said you can only fuse one of them at a time? And so to test that, we took our 3 best activation domains, which are actually kind of known as the only thing that actually worked from the whole screen. And that was VP64, P65, and RTA. And again, in this case, I'm using that same fluorescent reporter assay. On this time, on the x-axis, I'm quantifying for you the fluorescence from that reporter. And you see when you have the reporter control alone, you don't get much activation. When you fuse each of the single domains to DCAS9, you get a little bit of activation, but nothing outstanding. When you fuse them in pairs, though, you get a nice bump in activation. And when you fuse the whole triple together, you get quite a nice difference. It's about a 10-fold difference between the triple fusion and VP64 alone. And so at this point, we thought these data looked pretty promising, but it was just on a synthetic reporter. And so we weren't sure this would actually translate to endogenous targets. So to test that, we went after the VEGF locus. And this time I'm quantifying for you VEGF levels in protein on the x-axis. And you see each of the single fusions give you a little bit of a bump in VEGF. The double fusion looks even better. And again, the triple fusion looks the best. And again, about a 10-fold difference between the previous gold standard VP64 and our improved fusion VP64, P65, and RTA. And so for the remainder of the talk, I'm going to call this activator BPR after the constituent parts that make it up, or I'll sometimes call it Viper. And frankly, I just do that because it sounds cooler. So other genomic loci that we tested. So we tested a bunch of different sites. I'm only showing you a small subset of data. NeuroD1 is a transcription factor involved in neuronal differentiation. And again, when you use VP64 to turn it on versus Viper, Viper looks much better. ROXF2 is a transcription factor involved in spermatogenesis. And again, Viper turns it on much better than VP64. About 20-fold difference between the two technologies. And today we've tested a lot of loci, and in general, Viper tends to be superior to VP64. And so activating one target at a time is nice, but where I trained into my postdoc was with George Church, and George is really into multiplexing. Basically, if you're not multiplexing in the lab, you're not cool. And so reason why multiplexing in this case actually is important is that the way that the cell actually actuates its functions is not just by pulling on one lever at a time. When the cell sees heat or the cell sees DNA damage, it doesn't just say, oh, let me turn on one gene to solve this problem. It turns on whole networks of genes. And if we ever want to be able to sort of model that complex biology, we need to have tools that let us pull on multiple levers. And so we decided to test whether or not this technology could be used to turn on more than a single gene at a time. And so to test multiplexing, we targeted 4 genes at once. These are the 4 genes. One of them is a link RNA, MIA, and the other 3 are transcription factors. Y-axis is quantifying the fold RNA expression. And again, VP64 certainly does turn these genes on, greater than the control. But when you throw Viper in, it's a much bigger improvement. And again, about a 10 to 30-fold improvement in Viper over VP64. So summary for part 1 of my talk. We designed a new synthetic activation domain. It shows about a 10 to 30-fold improvement over the previous gold standard. It works great in human cells, and I've shown you pretty much only human data, but I've also done a lot of experiments in mice cells and fly cells, and actually even in yeast cells. And Viper is actually better than VP64 in all those contexts. And in yeast in particular, it's actually pretty incredible because this tool was evolved to work in humans, and yeast and humans are separated by about like 800 million years of evolution. So it's kind of impressive that this thing still works in that context so well. We also, in the original story, used Viper to turn stem cells into neurons, kind of showing that you could use these CRISPR-based tools to do programmed cellular differentiation, which is one of the goals we originally had. And since this publication, we've been really happy and surprised to see that it's been adopted quite well by the community, and about 1,000 people have asked for it through via Adgene. So it's kind of nice to see this tool proliferating and seeing all the cool things people have been doing with it. So that was our kind of our work on activators. I'll now switch to tell you a little bit about more recent work on repressors. And so again, why should you care about repressors? Because you just cut everything? Well, cutting still has the same downsides it always had originally. In addition, there's some areas where I think repressors are particularly useful, which is when you're going after things like long non-coding RNAs, because in those cases, there's no real reading frame to disable. So coding genes are nice that if you cut them and you just knock them out of frame, you're probably going to disable the gene or at least make it not very happy. But with long non-coding RNAs, there's really no frame to knock out. And in addition, in most of them, we don't even know what the functional region is. So you don't know really where to even begin to target. And so we really think that repression is a nice tool in those contexts. In addition, sometimes you need to induce partial loss of function because full knockout is not basically lethal. And so in the cases of essential genes, that's really obvious, right? If you knock out an essential gene, it is essential, so the cell just doesn't like it. And so again, a partial knockdown via repression might be a better tool. In addition, repressors are nice because you can kind of also get allelic series, just 10% knockdown, 20% knockdown, and so on and so forth, to kind of see the gradient of activity the protein can do, because at different levels of knockdown, you might see different phenotypes. And so when we started this project, what was the current state of DCAS9 repressors? So again, we weren't the first people to think about this, and these tools certainly existed and they certainly repressed, but the repression wasn't complete. And so this is from one of the original papers trying to target CD71. On the y-axis, they're quantifying for you the relative RNA expression in the cell. On the x-axis, they're showing you either the negative control or cells that got 1 of 3 different guide RNAs. And when they throw DCAS9 into the system, they do see some moderate repression where if you have DCAS9 plus a guide, you do kind of knock the gene down 40% or so. And they then tried fusing this crab repressive motif to DCAS9 to see if they could improve the repression. And they did. Again, certainly it helped, but this was far from perfect. And so we thought to ourselves, well, is this as good as it gets? And kind of when we started thinking about this project, we had just completed working on Viper. And so we had this whole experience of kind of building a better tool. And we said, could we take the tools and the knowledge we gained from building Viper and just apply it to a different problem, building a better repressor? And so what was our experimental setup? Very similar to the original one: identify repressive domains, fuse them to DCAS9, test them on a fluorescent reporter. In this case, the reporter is a little bit different in that it has a constitutive promoter driving YFP. And so the cells are normally yellow. Then if the DCAS9 repressor binds to it and is able to shut down transcription, the cells will turn dark. And again, we just quantify this using fluorescence, flow cytometry, I should say. And so what did our screen look like? On the y-axis, I'm quantifying for you median fluorescence intensity off of that reporter. X-axis is everything I tried fusing. And given that Viper was kind of a mess, I tried fusing a few more things this time to see if I get more shots on goal. And so when you have the reporter alone, you get kind of an activity about 100,000 arbitrary units from this reporter system. You add DCAS9, it kind of drops by about a log. And if you fuse on all these different domains, you basically get kind of different responses from each of them. And there are 6 domains that look particularly promising over here, colored in blue. And so based on Viper, we kind of knew that combining these domains might be the way to go to get kind of like synergistic repression. And so we did an additional screen in which you did all pairwise combinations between our top 6 repressors and the crab domain, which is kind of the gold standard at that time. So that's basically 49 constructs tested on the same fluorescent reporter assay. So to remind you, the cells are normally yellow, and they'll become dark if the repressor works. And so on the x-axis, I'm quantifying for you median fluorescence intensity. On the y-axis, I'm showing you all the different pairwise combinations we tested. Again, the reporter alone gives you a value about 100,000. You add DCAS9 in, it drops by about a log. And this is what all the pairwise combinations look like. And so at the end of this, we had kind of 5 domains that look pretty promising. And I'm sure everyone right now is thinking to themselves, you know what I'm going to do next, which is basically I'm going to do a triple fusion. And again, following the whole idea of Viper. And you'd totally be right. We definitely tried the triple fusion, but sadly it didn't do anything. And so that was somewhat surprising. And so basically we kind of went back to our original double fusions and just characterized them. And so long story short, we kind of played around with quite a few of the different double fusions and found the best one. And the best one was this DCAS9 fusion to the crab domain plus this MECP2 repressive domain. And so now we decided to test crab MECP2 against crab alone. And so we targeted the CanX locus as just an arbitrary locus to target. On the y-axis, I'm showing you relative RNA expression. We've normalized everything to the negative control. So that's always gonna give you a value of 1 in the data I'm gonna show you. You add crab to it, you get about a 40% drop in transcription. And you add crab MECP2 and you see a nice increase in the repression. And it's about a 2 to 3-fold change between the 2 tools. And again, if you target other loci, again, you see about another 2 to 3-fold change. So kind of crab MECP2 just consistently being better than crab. And similar to Viper versus VP64, sort of crab MCP2 sort of beats the previous gold standard across the multiple loci. I'm just not showing you all that data. And so again, we also tested this in a multiplexing context because again, we kind of realized that the way that the cell does things is not by pulling one lever, but it's by pulling multiple. And so we want to always make sure that the tools we build can pull those multiple levers. And so to test multiplexing, we targeted 4 genes at once. Again, y-axis quantifying new RNA expression level, and the x-axis are each of the different genes we're going after. This is what crab does for them compared to the control. And this is what crab MACP2 does. And so again, it's about a 2-fold to 3-fold improved repression with our tool versus the previous gold standard, and showing that it still operates within this multiplex repression context. And so you might be thinking to yourself, well, 2-fold repression like sounds like a better number, but does it actually have meaningful differences when you're doing your experiments? Is actually going to help you. So to test that, we decided to put the crab MECP2 against crab, sort of in a head-to-head competition in a mini screen. We had a library of about 1,000 guide RNAs, and they were targeting either 50% of them were targeting essential genes, which I'm showing you in this diagram as colored with orange cells, or 50% of the guides targeted non-essential genes, and that's the purple cells within this diagram. And so, and if essential gene is targeted and is properly repressed, you expect those cells to not grow that well, and so you expect them to deplete over time in the screen. And so basically the orange cells you expect to go away over time. And so that's exactly, so then when you passage the cells, essentially at the end of this, if your repressor is really, really good, you should only be left with purple cells. That's kind of how the screen works. And so what does the data actually look like? So when you look at DCAS9 crab, the y-axis, I'm quantifying you the full change in the guide RNAs at the end of the experiment, which is at 14 days, versus the beginning of the experiment, which is day 0. So after 14 days of this repressor kind of trying to silence this gene, how does that guide RNAs abundance look like? And so on the x-axis, I'm going to show you all the different genes we're trying to repress. And essentially, every single dot here represents a different guide RNA against a different non-essential gene. And so what you see is that sort of all the different numbers kind of cluster around 0, basically showing you that when you target a non-essential gene with crab, you don't really see a depletion of those cells. And that kind of makes sense because you're not targeting anything the cell really needs to proliferate. While if you do use crab to target an essential gene, you do get a little moderate shift, and that you kind of see there's some stuff that's going a little bit further down, but doesn't look that different from the cells that had non-essential guides inside of them. Now, if you compare that to crab MECP2, and again, I'm going to quantify it for the same way, where the y-axis is the full depletion, x-axis is different genes. If you go against a non-essential gene, you don't really see much repression. In general, most of the cell guide RNA abundances are kind of floating around 0. They're not really showing enrichment or repression or depletion. But if you target an essential gene, you see quite a marked depletion in guide RNA abundance. And so that's particularly surprising because if you look at the y-axis, they're basically different by a log between these two tools. And so crab MECP2 is dropping it by about 1,000-fold, while crab alone is dropping these guides by about 2-fold. And so that's quite a nice difference when you do your screen. So summary for part 2 of my talk. We designed a new synthetic repressor, which is this crab MECP2 tool. It beats the previous gold standard crab tool. It helps increase your signal you get from screens, which is important because you want to make sure you're getting all the hits you can out of it. And in data not shown, it actually enabled us to do sort of gene-gene interaction screening. When we did the same set of pairwise knockdown using the previous technology, the screen just looked like a total mess. We couldn't make anything out of the data, but when we used our enhanced tool, we actually could see signal and start to see genetic interactions we would expect between our different genes. In addition, using this tool, we were actually able to do quite complex, build very complex synthetic circuits that previously weren't possible with the previous technology because they just weren't robust enough to repress the different pieces of the circuit. And again, we've been lucky enough that this tool has been adopted by the community. And again, it's really great for us to see it being used in all sorts of nice ways to help further science. And so kind of what are the future steps for our lab? We've built these 2 nice tools and we're kind of happy to see that people have embraced them. But these tools, while I think are rather nice, they do have some limitations. And that really not every single guide RNA against your gene of interest works. And so ideally, you'd really want that because, I don't want a person to have to go and test 4 different guide RNAs against their target gene to know that hopefully one of them works. It'd be great if you could just pull any guide RNA off the shelf, throw it into your cell, and just feel confident that it would repress. And right now, the technology just can't do that yet. In addition, our tools are actually made with either rat parts or viral parts. And in the case where you might want to one day use these in vivo therapeutic contexts, that might be immunogenic. And so you might not want that. And so we're kind of seeing if we can replace some of these non-human parts with entirely human parts. And so to date, our lab's actually built an entirely different way of exploring and building these tools. We sort of have this combinatorial protein engineering system that lets us sort of build thousands and thousands of different Cas9 variants to test them all at once. And so we're now using that system to try to discover improved CRISPR activators and repressors. And hopefully I can join you maybe in a couple of years and share that data with you. With that, I'd like to thank a few individuals. So, I'm basically nothing without all the great people I've been able to work with throughout my life. For the activator work, I worked with two very talented people in the church lab, John Scheinman and Sue Vora. For the repressor work, I worked with Nancy Yu, another talented postdoc in the lab. And I also like to thank the current lab members that we have in our kind of new lab over at Columbia University, because they make going to work awesome and really make me happy to be a scientist. And obviously nothing exists without funding. And so it's really great that we've been lucky enough to be able to get some funding to continue our research. And with that, I'll take any questions.
Thank you, Alex. Any questions from the speakers to Alex?
I have a question about your screen. So is it guide RNA design, or is it the locus and sort of permissibility for repression? Can you talk a little bit about that?
Yeah, I think it's both, to be honest with you. And it's really hard to deconvolute those without testing like a lot of guides RNA against any given locus. But it's certainly true that sometimes at one gene, all 4 guides just work amazing. Another time at a given gene, 1 guide works well. And is it that the guide RNA just stunk? Is that there's a bunch of chromatin there and histones blocking it? And I just think I don't have enough data to comment like solidly and explain that phenomenon yet.
So have you tried like with a guide that didn't work for activation or repression to see if you pair it with a nuclease, would it cut?
Oh, interesting. I have not done that experiment. That's certainly, yeah, I haven't looked at the correlation between those two, and that would be an interesting correlation.
Because, I mean, from what we experience, the majority of the designs work as nucleases. But the percentage of guides working for as binding components seems to be much, much lower.
Yeah, one thing that's interesting is that no one's really done like so there have been tons of high throughput screens characterizing cutting and cutting efficiency. I'm not really aware of someone doing the same sort of thing for binding at scale. They're trying to figure out like, what are the rules for good binding? I guess Prashant Mali's original BP64 paper kind of looked at that, but that was really only like 1 permuted locus. But yeah, interesting question.
Okay. I have a couple questions here. This one's from Jeff Miner. You only barely touched on using multiple guide RNAs for the same gene to activate or repress, but can that increase efficiency?
Oh yeah, so 100% it does. So for activation, it works really well. So one of the reasons why we're still looking for better activators is that I know if I take the current activators and do 3 guides against the same gene, it'll go up like a log over any single guide alone. So I think there definitely is some benefit from it. For repression, at least in our hands, multiple guides does help, but in our hands, it seems to be that basically you're as good as your best guide in the repression case. So we don't seem to be seeing synergy, but there's some conflict. It's like some people in the literature do see synergy. So I'm not quite sure the difference is between us, but there's debatable in that case, but certainly it doesn't hurt you.
The second question here from Michael Rochman. Can you comment on technologies for evaluating distal regulatory regions or enhancers?
Yeah, so I think there's, so 2 technologies come to mind for me. There's the group that kind of does tiling nuclease activity. So you just kind of take your upstream region and you basically pound guide RNAs against it, cutting, and you basically look to see when you cut the locus, is there like an enrichment in the output of that gene essentially. The other method that people use is they basically just tile dCas9 crab in the same sort of fashion. And so again, you're basically just looking for where can you put a CRISPR system that then causes a change in signal. And by doing that, where you see the change in signal is where you impute there to be a regulatory element. Those are kind of the current existing CRISPR-based methods. Obviously, there's other reporter-based systems where you just like order oligonucleotides and clone them into reporter vectors and then test them that way.
I have a quick question. So it's interesting you mentioned like you are doing the screening like different Cas9 variant. So what's the hypothesis behind that? Do you think that different variants might play a different role for like activation or repression, or the domain or the guide RNA will be the main vector?
Yeah. So sorry, I should have been a little bit more clear. So I'm not testing different variants of Cas9. I'm still using just vanilla SP-Cas9. What I'm doing is I'm permuting the activation domains fused to it. So my original work, I took 3 activation domains and permuted them. In the later work, I took 7 repressive domains and permuted them. In the case what we're doing now, we have like 100 domains that we're permuting essentially. And so we think that there's going to be some optimal combination within that.
We will hear from Luke Gilbert later today about also repression. I just want to see with your configuration, how long does the repression and activation last?
Yeah, so that's actually something that we've never really looked at, to be honest with you, and we should. I think the idea of hit and run epigenetics, the fact that you could do a transient pulse of a CRISPR tool and get a long-term change in state, I think is super, super interesting. And I'm glad Luke's here. Luke has amazing data towards that, which I think will excite people. And it's something that, again, we really do need to look at for our own tools, but we just haven't.
Here are 2 more questions from John Birmingham. Did you test VP64 versus VP16 in VPR, since in some contests, size is important? Did you look at the different minimal promoters?
I did not try, Max, is that right? So I've probably tried like 100 variants of Viper actually that I just don't talk about to people. I think I actually might have tried that, but it's been so long, I don't remember the data. One thing I can say is it clearly wasn't massively better or else I'd be using it. But yeah, I think, I did try a lot of things. I tried changing linkers, going after different genes, permuting order, adding extra NLSs, adding other domains. Basically, I could get a version of Viper that was about 2-fold better after like all that testing, which is just unpublished because I didn't think it was really worth it. But I do not remember off the top of my head the direct answer to your question, sadly.
All right. Thanks again, Alex.
Okay, thank you.
Aaron is currently an associate professor of physiology at Medical College of Wisconsin. I have known Aaron for a long time. He is credited for creating the first knockout rats using zinc finger nucleases in 2009. And he was awarded a new Innovator Award from the Office of the Director of the NIH for that. In addition to his own research, he also runs the Rat Transgenic Service Center, which has a repository of hundreds of knockout rats made by ZFNs, probably also talents, and more recently, CRISPRs. So my hope is really just to let more people know of this valuable resources. And if you're thinking about using rat for your research, maybe Aaron already has the rat for you. So his title is Engineering Rats and Rat Model Resources. Aaron?
Thanks, Joshua. That was a very kind introduction, and it's good to see you again. Known each other for a long time. So thank you again for inviting me. This has been great. It's great to have a variety of topics in this morning's session already. And my lab's focus, as Joshua pointed out, is really on gene editing and genetic engineering is my main drive in a laboratory and my science. But we apply that the technologies that we use to many different areas, including I got to get my mouse on the right screen here. Including trying to uncover different mechanisms of disease, building interesting animal models for things like xenograft studies, and a big part of what we do is applying our technologies to build resources, which is something that I'll talk about today with you guys. And so I don't think I need to tell everybody how we apply CRISPR and ZFN and TALEN technologies to rodents through typically pronuclear microinjection. But here are, as Joshua said, we were involved in a collaboration with Sangamo Biosciences and Sigma Aldrich to show for the first time that these technologies at that time, zinc finger nucleases, be applied to an embryo to create targeted gene knockouts in the rat, which had not been done before because rat embryonic stem cells that were germline competent did not exist at the time. And so that, of course, was a big breakthrough for a big field of researchers who use the rat as a model. And so, of course, we try to take advantage of these two very well known and widely used pathways of repair for generating gene knockouts and gene knock-ins through the homology-directed repair pathway. And lots of models in the rat have been created this way. Something that kind of separates, I would say, or is probably more prevalent in the rat community than the mouse community, is the use of different inbred strain models, which have very unique disease model characteristics. So I would actually divide the rat model research community into two camps. One group of researchers who basically uses outbred rat models, rat strains like Sprague-Dawley or Wistar, or perhaps Long-Evans. We see these types of models a lot in the behavioral research field, also in the pharmacology and toxicology kind of fields. And then there's the group of researchers who use genetic models, so inbred genetic models, which have been selectively bred for interesting traits. And there's a large variety of these models and resources out there in the community. And so when we, in the laboratory, one of our focuses is really taking advantage of the broad phenotypic and biological differences in these strains to use our technologies and apply them to lots of different models, lots of different disease models, and generate some new tools and new animal models for a whole variety of disease areas and of interest. And of course, we're interested. We make lots of knockout models. They're rather trivial to make knockout rat models. Knock-in models, I'll talk about some of the challenges we have there. And of course, we're working our way to continue to work our way. One of the areas we struggle with is generating floxed models. And so I'll talk a little bit about that. And I look forward to Xiaoshu's talk coming up next. And so we've had a lot of success applying zinc finger nuclease. We did a little bit of work with TALEN technology. We didn't, at least in our hands, the mutation rates weren't as high as with the ZFN. And just about the time that we were getting frustrated with that, CRISPR, of course, came along, and we've done most of the work since then using CRISPR-Cas9 based technologies. As I said, generating knockouts is quite efficient. This is sort of the mutation rate in terms of genetically modified founders. The project success rate is well over 95% for generating, especially knockout models. The knock-in models, where we're trying to take advantage of that homology-directed repair pathway, I think people experience this in general in most model systems, where the HDR pathway is much less efficient than the non-homologous end joining pathway. But it is a big challenge. Things in general are less efficient in rats than they are in mice. And so if you have another drop in efficiency, it does make generating knock-in rat models quite a bit more challenging. We use a different kind of tool, a jumping gene tool, when we want to make transgenic overexpression models. And so that's, we use the Sleeping Beauty transposon system for that, and it's quite effective for making transgenic models. So, one of the big drivers of, I think, some of the challenges with efficiency is not just the species, but we're often tethered, to the biology of those disease model strains. And that's where we want to do our genetic engineering. And so sometimes those models are not great for the transgenic core. They don't produce lots of embryos. Maybe their embryos don't survive as well after you poke them with the needle. But that's where we're focused because that's where the biology is. The genes that we want to investigate are in those models. And so we tend to focus on trying to improve the ability to get embryos and improve the viability of those embryos for manipulation. So we've applied, having the ability to, being among the first to develop knockout models and having the ability to do this do it at a scale, I think that most laboratories, at least laboratories that do rat model engineering, can't do. We have sort of the dedication of a large team and a pretty good sized transgenic core, where we can really devote 4 or 5 days a week to injecting rat embryos when needed. And we've gone through now just a couple of, I'll touch on 2 of sort of the bigger rat resource projects that we've had in the lab over the years. And the first of these was a grant that came out of the American Reinvestment and Recovery Act funding. It was an opportunity just sort of on the heels of publishing the first knockout rat story, where we were interested in trying to translate human genetic association studies into a sensitized rat model. And so our goal was to develop a whole bunch of rat strains that would be knockouts for genes that were candidates for a particular common complex disease in humans. And we were really focused on hypertension and chronic kidney disease. And so the platform for this was, we wanted to take advantage of the phenotypes of a particular strain of rat called the Dalsalt sensitive model. This rat is an inbred strain developed many years ago and used in a lot of literature in the area of hypertension research. And it has this very nice phenotype where if you switch the rats from a normal salt diet to a high salt diet, they have this, within days, they have this sort of rapid increase in blood pressure, which, achieves a clinical hypertension quite rapidly. And along with that increase in blood pressure, we see sort of a time-dependent increase in kidney injury as evidenced by the excretion of albumin in the urine. This model has many features that are commonly plague humans with hypertension and the long-term disease consequences of hypertension. And so we think it's a good model upon which to manipulate genes to understand the mechanisms. And so we've approached this model over the years in a couple different ways. One was a project where starting in the early 90s to use mapping, so linkage analysis, to identify what is the genetic basis of the salt-sensitive hypertension phenotype in the SS rat model. And I won't talk about those studies today, but what we were trying to do in this particular program was to start to get after the question of how do we start to unravel this complex picture of human genome-wide association studies. So if you've seen a plot like this, you know that these are the different chromosomes. Each color represents a different disease phenotype or characteristic that was studied. And then each little dot or lollipop is an association between a particular locus and that phenotype. And so for any phenotype and any locus, there is a human, haplotype, which is a series of SNPs in linkage. And those haplotypes may include one or even dozens of genes. And so the question becomes, in a complex locus like this, the human genetic mapping only gets us to the haplotype. What is the, which of the genes is actually responsible for the phenotype? And so when you have a tool like zinc finger nuclease technology, you can start to tease this apart. And so the idea was taking the ZFN technology and applying it to our dull salt sensitive rat model and looking for gene knockouts that would either exacerbate or rescue the salt sensitive phenotype of this model. And so when you do that, you expect that not every gene you pick in these regions is going to have a phenotype, and that's exactly what we saw. So the majority of strains, we knock out the gene. There's no effect on blood pressure. There's no effect on urine albumin excretion. But then certain genes, when knocked out, can either exacerbate or attenuate the hypertension phenotype in this model. And so these then become candidates for further investigation. And so you can imagine that a haplotype that's simple, and maybe the entire haplotype here, which we could see sort of in this region where these SNPs are associated, may be contained within a single gene, and knocking out that one gene can have a dramatic effect on the phenotype of this model. And so we find that that's a good way to kind of validate the human GWAS approach, is that when we manipulate the genes, we see a phenotype that's clinically relevant. Of course, other regions of the genome, perhaps where there are several genes in the associated haplotype, we can find that they are actually much more complicated. And so knockout of some genes has no effect on blood pressure, but knockout of, for instance, the NPPA, which encodes atrial natriuretic peptide, increased blood pressure, while knockout of this chloride channel, which is the neighboring gene, actually decreased blood pressure in the SS rat. And when we take a look at the albumin excretion, we see even more complex interactions. And so underneath these haplotypes that are associated in humans with chronic kidney disease, diabetes, hypertension, some of the major things that plague us in our healthcare systems, there can be more than one gene potentially that are playing a role. And using this knockout strategy in sort of a high throughput in terms of making knockout animals and phenotyping them, doing it in that way can start to uncover some interesting biology. Some of the anecdotes that we've learned along the way about making knockout rats is it's not simply recreating something that already exists in the mouse. Sometimes there's actually real interesting biology that you can learn by making a knockout rat that you did not know when you studied your knockout mouse. And the phenotypes can be quite different. So one of my favorite examples is the APC knockout mouse compared to the APC knockout rat. Both of these particular models are ENU induced, but there's a very different phenotype, both in the distribution of tumors, whether there's a gender bias, the role of hormones in these models, whether they capture other phenotypes that are sometimes seen in patients that maybe the mouse model did not capture, and of course the larger rat model sometimes provides you some technical abilities that are very difficult or maybe impossible to do in a mouse. And so there's a couple other examples where having the knockout rat model can capture sometimes the clinical phenotypes that may be missing in your mouse model. Another one is the example is the Fabry rat model. So both of these models is a knockout of the GLA2 gene, there's an accumulation, or part of the disease phenotype comes from an accumulation of alpha-galactosyl. And so both models actually recapitulate that. But the mouse has a very mild phenotype. And this has been used, and different laboratories have used this model, and even companies have tried to use this model to develop therapies for Fabry disease. But the rat model, Fabry knockout, has a much more severe phenotype and actually has a reduced lifespan. And so sometimes making the rat knockout is not simply, duplicating something that already exists in the literature, and you can actually create something that's quite useful and powerful. The second sort of large rat resource program that I wanted to talk about today is the Genome Editing Rat Resource Center. And so this is a model where we actually sought to extend our capabilities of generating rat resources out to the research community. This is a project where we solicited the research community to submit applications for making rat knockout models. And so the idea was we would make your knockout rat or transgenic rat for free. And so the idea was you have to apply to the program. Your application, and because it was NHLBI funded, has to do with heart, lung, and blood disease research. There's an actual scientific review, and the external advisory group that reviewed those applications would tell us which models were scientifically meritorious to produce. And so this program ran for about 6 years. We received and solicited applications from around the globe, of course, most of them here in the US. We had about 200 applicants, very high success rates of getting your model made. And we generated a lot of different rat models. Again, most of them the knockout, many transgenic, a few back transgenic models, and a few of the knock-in, and one successfully fluxed rat model. So reasons, but not every project was successful. And so reasons for that is that sometimes the background strain was too difficult. You couldn't get enough embryos. Of course, sometimes you run into issues with the transgene, or the mutation itself can be haploinsufficient. And there was a sort of a resource limiting aspect where, following many rounds of attempts at producing some of the more complex models, we basically just had to throw up the white flag and say that some of these models, such as the fluxed rat models, are just too difficult to produce it in a sort of a high throughput way. And so, this issue of, generating global knockouts is, again, quite easy by pronuclear injection of embryos in the rat. But, getting at the challenge of generating more complicated models like the knock-in or flox models is something that continues to plague the rat field. And so, the rat embryonic stem cell technology is I think, a viable alternative. We're getting better at developing and having ES cells from rat strains that are more important for a broad variety of researchers in the field. So I think this ES cell technology, while viable, is really, I think it was slowed down quite a bit because of the use of CRISPR and the pronuclear approach. But I think there are a few researchers, including ourselves, who are trying to get rat ES cell technology up and more mainstream. We're looking into newer approaches for delivering CRISPR and other tools to rat embryos. So getting away from microinjection towards using electroporation. I think we're going to hear a lot about that in the next talk. One of the things that's going to help this are some very new advances in rat embryo culture. So one of the other main challenges to working with rats in the transgenic core is that the embryos are often difficult for many strains to get to culture past the 2-cell stage. And so there are some new advances in rat embryo culture, which we think are seen to improve this in some of the inbred strains that we work with. We're of course very interested in some of the even newer approaches, combining site-directed nucleases with AAV technology. And there's also potential to use other technologies such as spermatogonial stem cells for sort of large-scale engineering of the rat genome. I've made it my 2021 resolution for my group that we're going to try as many different strategies for floxing genes in the core as we can. Of course, the thing that we battle against is when trying to insert LOXP sites using CRISPR or ZFNs, is that there's a lot of potential outcomes on an allele basis of what you can find in your founder animals. Quite often you end up with, when you're trying to cut on both sides, you often end up with a deletion of the target exon or your LOXP sites being in cis, or you can have a LOXP site on one side and an indel on the other side. And only very rarely, at least in our hands, do you get this in cis. I'm sorry, before I said in cis, these would be in trans, but very rarely do you get the in cis configuration of LOXP sites. So I think Xiaosha is going to tell us how her group does this quite effectively in mice. When we do have the ability to generate these kinds of flox models, we are ready to implement them. So one of the things that the resource programs that I'm involved with and my own laboratory has been doing is developing the Cree tools, the Cree strains for rats. And there are many other groups around the country working on this. One of the nice models that we take advantage of is another transgenic knock-in model, very similar to the AI9 mouse model that you heard about in the last talk, where this transgene is inserted in the ROSA 26 locus and it has the stop cassette upstream of TD tomato. And then upon Cree activation, you get this transgene expression. And I'm not sure, Xiaosha, if you were involved in actually making this rat, but it's a very useful strain made by, at the time at Sage, and then we breed this model at MCW now. And so one of our areas of focus is the kidney. It's an important organ in the regulation of blood pressure. And so our group has been developing the endothelial specific Cree, podocyte specific Cree, proximal tubule, and collecting duct specific Cree models. And we can show that when we combine them with the reporter strain, we get this very nice tissue specific activation. And so once we have the ability to more reproducibly flox genes, we're going to be able to leverage those tools. I'm wrapping up here. And so if you're interested in finding a rat model that we have sort of on our shelf, you can visit the rat genome database website at rgd.mcw.edu. This is a great resource for anything involving rat, curation of the genome and all of its parts, as well as strain model resources and phenotypes. And so you can come here, and if you click on this button at the top, it'll bring you to our models page, where we have a listing of the strains that are basically currently available, and we try to update this periodically as new strains are added. And so I encourage you, those of you who may be interested in a rat model, to come look here or reach out to me directly to see if maybe we've already produced it. And with that, I will stop and thank all of my collaborators, my group here, both present and past members have all contributed to this work. The Genome Editing Rat Resource Center, my collaborators there, Mindy, Howard, and Joe, and of course, Mindy's team, who continues to develop and sustain these resources. We couldn't do this without the our collaboration with the MCW mouse transgenic core, and they're two excellent technicians who do most of our embryo injections for us. Of course, my collaborators in my department, and all of the rat aficionados out there who keep us very busy producing new models. And of course, none of this would happen without the generous support of our funding agencies. And with that, I will stop and take any questions.
Thanks, Aaron. Anyone in the speakers have a question for Aaron? I will ask the first one then. You mentioned a lot of people, at least some people, are still working on the rat ESLs. Mm-hmm. How many different strains are there, like, with ESLs developed?
I would say, so, right, so published, yeah, published is few. I think you could count the number of published germline ES strains on one hand. I think, you know, the Fisher, the dark agouti, maybe at that, and I think Qilong at USC has, I think he mainly uses a hybrid ES line. There's not many that are published. Our Akiko Takazawa from my group, she has developed ESLs from multiple of the inbred strains, like the dull salt sensitive model, the fawn hooded hypertension model, and she has those strains and shown that they're germline competent. We haven't published those results yet, and there are some groups in Japan who have developed several ES lines as well. So I think that probably the total number of different strains is still maybe on the order of a dozen, where maybe there's evidence of germline competent ES cells. The number of published examples of gene-targeted knockout rats produced this way, I think, is still under 5 out there in the literature. So it's still not a widely used and deployed technology.
You still think there is a need to make it more widely used?
If we can repeat the kind of success that you're going to show us here with generating flux models, you know, I think for us that's still the main hurdle, right? So we at least in a model like the dull salt sensitive strain, we just don't achieve the HDR rates in nuclear injection to make that as a viable approach. We often just have to quit. And so we are trying to see if, I think gene targeting ES cells is a viable alternative. It's not going to be a cheap alternative, but at the end of the day, hopefully you'll get your model. So it's sort of a backup plan, but I think if we can combine CRISPR with ES cells, maybe we can, and maybe some fancier gene trapping or other techniques, we could do this at a larger scale in ES cells than we can in embryos. But of course, we'd love to be able to stick with electroporation and pronuclear injection to produce floxed models.
I think Monica has a question.
I did. I noticed your success rate sort of for knock-in projects, it looked to be maybe about 50%. And I was wondering if that is, are those like larger cassettes, or are these just point mutations that you're struggling?
So are we referring to this?
Oh, that was 2%.
Well, right. So yeah, so this would be on the number of genetically modified, founders with sort of like a seamless, integration out of the number of founder pups that were screened. That's what this number represents.
Yeah, she was probably referring to
the other slide, actually. Yeah, with several projects.
Oh, here?
Boxed.
No, another slide.
Another slide.
Towards the end.
Yeah.
Oh, okay. I know which one you're talking about. Yeah, as far as the rat resource center. There. Yes.
There we are. Yeah, I was wondering if that's in like a relationship to point mutation or cassettes, like larger cassettes.
So all of these here were insertion of larger cassettes.
I see.
I think that I won't say all of them. I do know one of these was a knock-in of a point mutation, but I'm pretty sure most of these were insertion of reporter genes, large reporter cassettes, that just, after a certain number of attempts, we just had to move on to the next gene on our list. And of course, the Flox models is difficult. So that's the other area where I think either ES cells or combining CRISPR with AAV might get us over the hump with generating sort of those reporter gene insertions. I know this has become quite reproducible in the mouse model now, and I just don't think many groups have applied it to the rat yet.
Seth Henson has a question for the Flox project. Are you looking at utilizing any specific approaches to improve success rate, standard RNP, modified Cas options, tethered donors?
So we have done a little bit of work. We've tried some of the published methods for inhibiting non-homologous end joining. So there are some small molecule approaches to doing that. Some of our colleagues say they see a little bit of an improvement with HGR if you can inhibit non-homologous end joining. Nothing super exciting for us yet in that regard. We're trying some of the newer techniques. Prime editing is one thing that we're taking a look at. We have done things to like modify the donors to stabilize them, try to protect the donor molecule from being degraded. We looked at the timing of injections, 1-cell, 2-cell injections, and things like that. So far, and I think it's mainly because I'm quite stubborn, and I always tell my team, if it doesn't work in a dull salt sensitive rat, then it doesn't work. And that's mainly because that's where all of our funded grants are focused and in my department is studying this model, and we need to generate these models in that strain. So, we often go straight to the dull salt sensitive model to test a new technique. Some of these may work better in some of the outbred strains, which are quite robust. And there's certainly groups publishing. We're not the only group in the country, working with rats. And there are groups who are publishing different techniques, which seem to be quite effective for generating knock-ins. But my stubbornness is to work with these inbred strains where the biology that we're interested in lies. And so we needed to get these techniques working in those strains.
All right, thanks again, Aaron.
Thank you.
I will continue on this animal model track. Let's see, I need to share first. I don't see my screen. Do you guys see my screen?
I can see your screen. You just need to go to presenter mode.
Oh, now I see it. Thanks. Are you seeing my, just the slides, right?
Yep, you got it.
Okay, my title is Reliable and Efficient Creation of Flox alleles in Mice. Okay, so Aaron gave a really nice introduction to my talk, so that'll save me some time. Flox alleles is really one half of the components of a conditional knockout mouse. You need to insert LOXP sites into 2 sites into a gene. So a LOXP site is 34 base pairs long, like shown here. Let me show. You need to insert 2 LOXP sites into the same allele, so to flank 1 or more exons that after you, in the presence of CRE, the 2 LOXP the sequences between the two LOXP sites will be excised. So you will lose those sequences being flanked. So if those exons are encoding sequences that's critical to the gene function, or they encode coding sequences that's not the multiple of 3, then after excision, you will have a frame shift downstream, and which will create premature stop codons and lead to the messenger RNAs degradation through nonsense mediated decay. So this is an efficient way to disrupt genes only in the cells that CRE expresses, so that you can achieve tissue specificity or sometimes temporal specificity. So the method we're using to create floxed alleles is to design 2 guide RNAs cut twice, and the guide RNAs and Cas9 RNP are delivered with single-stranded DNA oligo for each side. So each oligo carries a LOXP side and flanking sequence. And so the SSODNs has this very simple configuration. You have the LOXP side. Usually we also include a restriction site there for easier genotyping. And we use about 6 nucleotides of homology arms on each side. And the whole SSODN carries 2 phosphatidylate bound at the very end of both sides. So our design criteria is really to look for exons, like I said, that's critical for function, or if we don't really know, we make sure their excision would cause frame shift downstream. And it's also important to take into consideration of splicing isoforms. So you want to make sure you take out the, after excision, all the splicing isoforms will achieve frame shift if that's what you're looking for. And you also want to use the same Cas9 variant. You don't want to mix your wild type with, say, VQR variant because the guide RNAs can't tell which one is which. So this method has its advantages. So it's very flexible. As I said, you want to insert 2 LOXP sites, but you are really looking for relatively two independent events. So you pretty much can insert them to anywhere in the gene. Sometimes it's not that critical if you have a fairly small exon and fairly early, that you can just flux and take it out and the gene will be degraded. Sometimes genes have many splicing isoforms. You will have to end up fluxing a larger region in order to make sure you take out the gene when in the presence of CRE. And so we just buy synthetic guide RNAs and other SSODNs with the modifications. It's relatively cheap compared to other method I would mention later. And most importantly, this method is really, it's compatible to electroporation of the embryos. As Aaron mentioned earlier, if you have to use a needle to poke the embryos, they don't always like it. So using 2 guide RNAs and 2 SSODNs to deliver LOXPs into a gene to make a floxed allele has been tried with different success rate. At the very beginning of CRISPR technology, the Yenish lab published a cell paper using the CRISPR plasmid and SSODNs to very efficiently create floxed alleles, but there's also a large group of labs that had not so success experience. But most of those work were done with a microinjection. Aaron also showed something similar. So with microinjection, you have to, under the microscope, using 2 needles. One is to pick up the embryo and hold it to immobilize it, and the other needle carrying the CRISPR reagents and poke into the zona here and into one of the pronucleus. And when you do microinjection, even though it has been established for decades now, the embryos, some of them would burst and some of them may not continue to develop. Anyways, with microinjection, usually you see much lower birth rate as a result. Alternatively, you can isolate the single cell embryos and mix it with your CRISPR reagent and load it into a cuvette and put onto your electroporator and zap it, and you can process many embryos at a time, and a microinjection session usually is 2 to 3 hours of very intense work. Surprisingly, even though you actually can see the reagents being delivered into the embryos, electroporation actually delivers reagents more efficiently, and more importantly, the birth rate usually is higher. A downside of electroporation is it really can't handle anything larger. So like a plasmid will not go in there, and Cas9 messenger RNA would be too big to electroporate as well efficiently. So Aaron read my mind. We have a very similar slides, as Aaron mentioned as well. So floxing is, well, it's not a simple editing. Even though for each LOXP site insertion, when you use an RMP with a SSODN, each individual site can be modified very efficiently in our hands. It's probably approaching 10 to 20% founder rate. However, when you want to insert 2 in there, there are just too many competing events going on here. You can have a floxed allele with 2 LOXP sites on the same gene and same copy. And you can have one of the site have an indel, the other side with a wild type, or one with an indel or a wild type, the other side has a LOXP. Or you can have embryos that just didn't get much your reagent, and both sides are wild type. And you can also have deletion. That actually happens very efficiently by CRISPR, is when you cut on the same gene, deletion can be obtained even when the distance is far. So when you are like the flox allele is competing with all these events, you just simply can expect floxing to be a super efficient editing. And so after the reagents are electroporated into the embryos will be transferred into pseudopregnant females. When the pups are born, we genotype the pups, usually taking a tail or a toe clip. So to genotype the pups, we are really looking just very locally at each insertion site of an amplicon of 250 to 450 base pairs. So we're looking for LOXP site in each side, essentially. But if you have a deletion or insertion that cause the either the primers of the PCR not priming to amplify, or the amplicons are larger than 450, usually it doesn't pick up by our NGS analysis. But all the others, if you have small indels, if the site remains wild type, if you have a LOXP site, it can be picked up very accurately with NGS, and it also has the ability to tell you how much of each genotype there is in the animal. What we cannot detect also is the large deletion, but often it can be implied in the genotyping data. I'm going to show you an example. So this is the readout of our NGS analysis. On the left panel is different animals, and the allele readout for site 1 and for site 2. As you can see here, even though we know for most genes, there are 2 copies, we can see some, that was a little, okay. We can see some of the animals actually has 4 different alleles at one side, and some of them just has 1 allele even on both sides, and a lot of them in between. So why is that? That's really just because we start with a single cell embryo for electroporation, but it really depends on how fast the cells divide after they receive the reagents and how long the RNP lasts in the embryos, and also how fast editing happens. If both of the alleles, when the cells is still, the embryos are still in single cell stage, were modified, then every cell in the animal eventually will have the same 2 modifications. And if one of the modifications is large deletion, then you will read every animal only has 1 allele at each side. But then if one wild type allele is left when the cell divides into 2 cells, you will carry 1 modification, which will be passed on to both cells, and the remaining wild type now has 2 independent genes to be modified differently. So you can, and if you have more than 1 allele, both alleles are wild type. When you go to 2 cells, then they have more diversity. That's what we're seeing in our genotyping. Of course, for FLOX projects, we want to look for animals that have LOXP sites in both sides. Shown here in this collection of animals, 2 males here, both have LOXP sites in side 1 and side 2. The top one here has a fairly low percentage of LOXP site at side 1 and a higher percentage on side 2. And the second animal looks like both sides only has 1 allele, and it's the LOXP allele, as I mentioned earlier. Most likely it happening first the single cell stage, and there's 1 allele that has a large deletion. So most of the time when we do a deletion PCR on those animals, you do detect a deletion. And for the animal on top that you have different percentage of LOXP sites, we really don't know whether we have some of the cells in the animal actually has a FLOXed allele, because we, with NGS, we are not able to tell the difference between an allele with FLOXed and a wild type and indel versus 2 alleles. Each one has 1 LOXP site at the 2 different sites. Then the remaining allele is wild type or indel. Usually you will have to breed the founder with 2 LOXP sites to the next generation. It takes about 8 to 10 weeks before you know whether or not you have a FLOXed allele. If you don't, you have to think the next step, how to either to repeat or something. It just takes a long time, given most of the time our animal cores have very long queues. It makes it highly unpredictable what do you need to do when you have your founders with 2 LOXP sites. So we started to implement this in vitro CRE assay that we developed quite a few years ago. The idea is when you look at the genotyping primers we use for each LOXP site, when excision happens in the presence of CRE, we will obtain a linear deletion allele, and then it generates a circle that's being excised. When it's circularized, you can see that the 2 primers that R1 and F2 used to be facing away from each other that won't amplify, now are facing towards each other. So this PCR will be specific to the excised circular product. And here's an example. We have 2 founders, both have 2 LOXP sites at different percentage. And the M32 actually has fairly low percentage. It's only about M45 actually has pretty low percentage, about 8% for both sides. M32 has a higher on one side, but a lower LOXP side percentage on the other side. So when Monica did an in vitro Cree assay, she used a control, which is 2 animals that only had 1 LOXP site for each site. So she mixed the genomic DNA on that for that control and the other 2 founders and a no template control. So what she did was she just incubated the Cree protein with the genomic DNA, then used that as a template to PCR amplify that circle specific amplicon. So for this, we're really trying to encourage the single molecule excision event, just in case to LOXP sites in trans would also give us a product. So we use fairly diluted DNA for the Cree assay. And so M45 had a positive PCR, M32 didn't, and this is confirmed in F1. So out of 11 animal of F1 offspring of M45, one of them has a floxed allele, and M32, even though it had multiple animals, each carry a LOXP, it didn't, none of them out of the 20 some animals had a floxed allele. And the 1 out of 11 kind of matched what we observed in the genotyping of the founders. So in the past 3 years, we completed 33 floxed projects. All of our flox projects are 4 sessions. We start with 4 sessions. Every session is about 100 to 150 embryos being electroporated. And the live birth usually ranges from 45 to 145. And among these 33 projects, the floxed range is somewhere close to 500 base pairs, all the way to 10 kb. And for the projects that we had multiple founders with LOXP sites, about 50% of the animals has floxed allele, and the rest of 50 that had LOXP sites on separate alleles or even in separate cells. All animals tested positive in Cree assays, transmitted a floxed allele in F1. And we also did some retro testing for the projects that had germline transmission of a floxed allele. When we went back to do the Cree assay, they're also all positive. We have 6 projects that's don't have germline transmission rate transmission data yet, but all of them had Cree positive founders. Some have multiple. So we expect them all go germline with a floxed allele. And out of these, we do have 6 projects that failed to transmit a floxed allele. Whenever this happens, you kind of have to think what to do next. Do you want to repeat the sessions? You could get another 4 sessions and this similar number of animals and not getting a floxed allele. And then the biggest problem is you don't know when and how much money you need to put in there to get a model. So we looked closer to the genotyping data, assume the same, this is the same NGS data. We had 2 potential floxed founders here, but what if we didn't have these? If you look at the rest of the animals, there are quite a few of them that had almost 100% at one LOXP side, but almost 100% on the other side with an indel. So we figured we can do something with the indel because this indel doesn't really, this genotype isn't present in the wild type. So if we take a male animal and the sperm from a male animal and do IVF with a wild type oocytes, we can get embryos that's hybrid for percentage of the animals with one allele that has one LOXP side, and the second side has an indel that's very specific to this allele, then we can generate another, we can just use another guide RNA that targets specifically to this indel with the original SSODN and re-electroporate. So only this allele with this indel will be targeted. The wild type allele will not be touched. So when we have another LOXP inserted in the F1 generation, they have to be floxed. So essentially, when we do 2-round targeting, we broke the more difficult floxed editing into 2 simple like insertion of 1 LOXP side. So timeline-wise, they're very similar. So essentially, you wait till the male founder with 1 LOXP side and an indel to be old enough that you can use them for IVF to generate F1s after electroporation. And if you already have a floxed founder that's Cre positive, you can just make them. And so to get it to the F1 stage, it takes somewhere around 20 weeks for both of them, both the strategies. But by the time with a 2-round targeting, by the time you get to F1s, usually you get multiple F1 with both LOXP sides, and we know they're already in phase. So this way, we kind of get a guarantee of like about 20 weeks, you will have a floxed allele one way or the other. So these are like 4 of the 6 failed projects were retargeted. These are the genotypes, so they all have 1 LOXP site in, the other one has an indel, except for the last one that we targeted a wild type second side. You can see the difference here just to show you how different these guide RNAs are. The top red one is the original guide RNA, and the bottom one is the new guide RNA. That the red dash shows the deletion in these, and we have to modify the guide RNA sequence accordingly. So again, we use the same SSODNs. So we have data for 2 of the retargeted 3 that uses a new guide RNA. And we got multiple pups with both LOXP sites. They are germline confirmed. And the ZFHX4, where we targeted a wild type site for the second site, we got 2 pups out of 40 with both LOXP sites. But when we do in vitro pre-assay, only 1 of them is in CIS. So that's understandable, but because for IVF to embryos, there are 2 wild type alleles to be retargeted, but the one that's in vitro Cre positive is germline, did transmit through germline. I just want to stress that importance of reagent validation. Even though nowadays with synthetic guide RNAs, most of the guide RNA designs are highly active, we always go through transfection of NER2A cells to make sure all the reagents are working. We very often see the cutting efficiency is over 80%, and LOXP insertion is over 10% in NER2A cells. We're doing this also trying to make sure we don't introduce any human error, whether it's from the operators or from vendors. And sometimes, very rarely, we do run into a guide RNA that just doesn't work. So the validation really, after you receive all the reagents, it takes a week. Usually our queue is much, much longer than that. But just a simple validation really helps to avoid repeating all the mouse sessions that are a lot more expensive. So there are other methods of doing floxing. You can use single-stranded DNA donor where both FLOXP sites are on the same donor. That kind of limits how much sequence you can flox because single-stranded DNA has a limited size you can synthesize, and it can be too big to electroporate and can be too expensive to synthesize as well, especially when it's very large. And there's another method called igonic. You can electroporate the oviduct of donor females with RMP and SSODNs. You have to use very, very high concentrations of reagents for that. And also if you need to retarget IVF, it's obviously not applicable with that method. I just want to very briefly talk about some new data using RAAV to deliver donors. So back in 2018, there were a couple papers published that instead of using plasmid donors, when you have a larger insert, they put the donor onto recombinant AAV vectors. And they just drop the AAV virus onto the embryos, then electroporate RMPs in there to also avoid having to do microinjection. And so far, we have completed 6 projects. The inserts ranging from 740 base pairs to all the way to almost 3 kb. And most of them gave us a fairly nice birth rate, and all of them gave us founders with both positive 5' end and 3' end junctions. And one of them even gave us 42 founders. It's way too many. But the good news is really like we can very reliably generate fairly large insertions with AAV as donor. It's a total change from what we experienced with microinjection. And we can even lower the homology arms to 500 base pairs for the standard 800 base pairs. That kind of gives us an opportunity to insert up to 3.7 kb as cargo in the AAV. We do realize that a bunch of projects had more or less random integration of the AAV sequences. We don't know exactly where they go into and what percentage of the viral genome got inserted, but it is potentially a caveat with this method. So I'm looking forward to hear the panelists' idea on how to detect and what's the consequences for model creation and for therapy especially. Just to summarize, I talked about floxing with 2 RMPs and 2 SSODNs. It can be fairly efficient. And with a second round targeting, we can kind of like, it's a very reliable method. And we use this in vitro Cray assay to analyze founders. So you can find out whether or not you have a floxed allele before you breed to F1 generation. And with all these, we can confidently say we can generate a floxed model in 20 weeks. And RAAV is a very efficient way to deliver donors without having to do microinjection for large insertions. I just want to thank most of the data I talked here is generated by Monica. She's gonna also lead the discussion panel later. And almost all the floxed models were created in Mike White's mouse core. And Mingjie Li has the Hope Virus Center viral core, and that prepped all the RAVs for us. And Mia Wallace has the other mouse core that contributed to some of the RAV projects as well. Thank you. I'm ready for questions. Okay. Fumi asked, is it possible that 2 LOXP sites are integrated into a different gene? I guess you're asking about random integration of single-stranded DNA oligo. I guess it's always a possibility, but a single-stranded usually has lower chance to integrate into the genome. There's really not a very good way to look for them other than whole genome sequencing the founders. We have saved a bunch of bodies. So at some time we will look into that as well. Aaron?
Hi, thanks. I'm wondering if you can comment on the mosaicism with electroporation versus pronuclear. So that's my first question. And my second question had to do with the AAV and whether you were using the self-complementary or the single-stranded AAV vectors.
Yeah, from what we have seen so far, even though it was kind of, it was surprising for me, electroporation actually delivers more RNP into the embryos. Of course, we are using higher concentration when we electroporate. I don't know how much that has to do with that, because microinjection, when you use too much, it's kind of toxic. I think with more reagents delivered into the embryos, that probably will reduce the amount of mosaicism compared to microinjection. And I also don't know whether, microinjection, after you poke the embryos, it makes them kind of in a shock a little, how that impacts repairing. We definitely didn't see more mosaicism with electroporation. With RAV, we are using single-stranded, not the self-complementary.
Okay.
Dongsheng, you have a question? You need to unmute yourself.
I have two questions. Sure. The first question said, for your AAV approach, it seems works very well. Did you try different AAV serotype or just one? We
used 6. That's what what's reported in there. But we, actually, we can ask you this question. We use 2 different, like, we use AV2 ITR. But we, some of the ITRs are identical, like they're just inverse. Some has some variants in one of the ITRs. We actually don't know why, but they both work.
They should be, and ITR can repair each other during the replication process.
I see. So we can
repair it. Yeah. So my second question is that the AAV do have the property of integrate into the genome. And so for your project, did you ever do a whole genome sequencing to see if AAV, when your donor integrates some places?
Well, we probably don't need to do whole genome sequencing because we know what's in the AAV sequence. For mouse, I mean, so we do a routine cassette PCR just to amplify the a couple stretch of sequences on the donor in the tail sample just to see if we have random integration. That's how those random integration were identified. But potentially, if we want to find out if elsewhere has a piece of the viral genome, we can just do PCR. What the nice thing about animal is you can always breed and lose what's potentially there. There are reports in the literature that most of the integration still contains part of the ITR. So I think we can probably use that to kind of design our PCRs to be specific to random integration, not the target integration locus. I don't know. Do you know if that's true, that most of the random integration contains ITR?
Yeah, actually there's a recent paper from Jim Watson's lab. They developed this ITR-seq approach, like guide-seq, they call it ITR-seq. So based on ITR to do a genome-wide screening of the potential integration of the AAV sequence.
Yeah. I guess I can ask you the same question. I mean, we use AAV very temporarily just to deliver the donor, but for therapeutic purposes, are you worried about random integration? You will have a lot more because of
it does happen, but it's happened the integration efficiency is very, very low, and recombinant AAV integration is not site specific. And also that it happened under certain circumstances, in certain models, it can get to the activate oncogene and cause problem. But this have to be like in a unique disease a specific model and sometimes specific age. You don't see it quite often, and occasionally you can gain benefit from integration. There's a study published recently that is showing that by doing hemophilia gene therapy with AAV, some of the dogs, their factor IX production went up after 7 years. It turns out that its integration, give more therapeutic protein in the end.
There's one more question. Could you comment on efficiency of the methodology versus electroporating guide RNA HDR as double-stranded plasmid? Electroporating. So this is asking about electroporation of SSODN versus plasmid. I don't think anyone has successfully electroporated DNA plasmid into embryos. I know they work very well in cells. We have not tried to electroporate RMPs and SSODNs in utero in embryonic brain. I guess the technology is out there. Do you think that RAAV can integrate as concatenators or not? I don't know. All right, let's move on to the next speaker. Our next speaker is Andrew Yu. He is Associate Professor of Development Biology here at WashU. He's instrumental in the direct differentiation from fibroblast to neuronal lineage. So his talk is modeling late onset neurodegenerative disorders via direct neuronal reprogramming of patient fibroblasts.
Thank you. Thank you for the introduction. And can you hear me well?
Yes.
Okay.
And we can see your slides.
Okay. So thank you for the introduction and the invitation to describe our work. So our work is mainly focusing on using microRNAs as factors of cell fate control, so that we can use these microRNAs to reprogram fate of cells from non-neuronal context to neurons. And we try to do this by this process called direct neuronal reprogramming. And the ultimate goal of using this technology is to model late onset neurodegenerative disorders. So we try to meet a number of challenges in terms of modeling disorders, late onset disorders. And these challenges are based on the recognition that aging is the biggest risk factor for most neurodegenerative disorders in humans. And is it possible to generate patient-derived neurons to study how the aging in the context of neurons and how neuronal aging contributes to the adult onset of neurodegeneration? And the story gets even more complicated because oftentimes neurodegenerative disorders affect very distinct types of neurons. A famous example would be cortical neurons. That are largely affected in the Alzheimer's disease and frontotemporal dementia, whereas in Huntington's disease is mostly striatal neurons, and within the striatum there's a specific type of neuron called median spiny neuron that are largely affected by the disease, relatively more so in comparison to cortical neurons. And I'll get back to the Huntington's disease later on. So while we try to meet these challenges, our key question is how to specifically and efficiently generate disease-relevant subtypes of human neurons. And can we use these neurons to study adult onset pathology of the disease? So my lab uses microRNAs called MIR9, MIR9 star, and MIR124. By the way, MIR9 star basically just reflects the fact that the two different microRNAs coming from the same precursor. So from now, I'll just refer to them as miR9 and miR124. So we use these microRNAs as reprogramming factors to be overexpressed or topically introduced in human skin fibroblasts. And we use these microRNAs as an effector to turn fibroblasts directly into neurons without going through stem cell stages. And this is why we call this process direct neuronal reprogramming or direct conversion, neuronal conversion. And the goal is to make skin cells, fibroblasts, directly turned into neurons. So let's start with the what microRNAs do. So microRNAs are small RNA molecules, usually about 22 base pairs in length. And they're initially transcribed as a prior transcript form in the nucleus. And the prior transcript is formed, it gets processed into the precursor form in the nucleus, after which it gets exported out into the cytoplasm, and they're further processed into mature microRNAs, about 20 to 24 base pairs in length. And what they tend to do is they tend to recognize their target transcripts, and they try to repress the expression of the target genes. And this repression of target gene may occur through two different mechanisms. One is the inhibition of translation, and the other one is the actual degradation of their target transcripts. And this inhibition occurs through different modes of targeting, whereas the mature microRNA here usually by the 5' regions of the microRNAs, they tend to recognize the target genes. But the thing is, it can occur in many different duplex formation. So the reason why we got into these microRNAs, miR9, 124, is the fact that I found these microRNAs to be a critical component of controlling the activity of remodeling, chromatin remodeling complex, BAF. It's called BAF chromatin remodeling complex. So what I found was that these two microRNAs are very neuron-enriched microRNAs that are upregulated at the onset of neurogenesis. Here you're looking at the mouse embryo cut around the developing neural tube. And what you're seeing here is that when this neural progenitors in green, when they differentiate into neurons, there's upregulation of miR9, in this case miR9 star, and 124. And what these two microRNAs do is they target this gene called BAF53A. So BAF53A is a component of chromatin remodeling complex called BAF chromatin remodeling complex. This is one of the subunits. But the interesting aspect about that is there's another chromatin remodeling complex subunit called BAF53B. So BAF53B is a subunit specific for neurons, whereas BAF53A is non-neuronal component of BAF chromatin remodeling complex. So in a way, these microRNAs act as a molecular switch by repressing the target gene, but at the same time instructing a larger complex molecule to be specified for the respective cell types. And another interesting aspect of these microRNAs is the fact that if you look at other targets of these microRNAs collectively, an interesting theme emerges. So here I'm showing you on examples of couple of examples of other known targets of microRNAs, and they're all different genes. BAF53 I just described, and there are also other genes called PTBP1, SCP1, COREST, and REST. They're all different genes doing different things, but they all share one thing in common, which is the fact that these genes that are labeled in red, they are expressed basically throughout the whole body, or many different cell types, but they are selectively absent in neurons. And what these microRNAs try to do is they try to recognize the genes that are not desired to be expressed in neurons, and they try to shut them down. So that was the rationale for why we use these microRNAs as a cell fate reprogramming factors, because if we topically introduce these microRNAs in non-renal context, so we use human skin fibroblasts as an example, will that happen? Will the conversion of fibroblasts towards the neuronal context will happen? by the virtue of these microRNAs being able to recognize the non-neuronal characteristics in human fibroblasts. So the original paper describing these microRNAs as a potential as a programming effectors, we published that a while ago. But here's basically a summary of what these microRNAs do. So when we topically express these microRNAs in human fibroblasts, and this is what the human fibroblasts look like in culture, and those cells basically turn into cells like that. And they look like neurons. They have the morphology of neurons, and they also start expressing markers that a lot of people typically use as markers of neurons. And these markers are like, for example, TOP3 or MAP2. These are more pan-neuronally expressed genes. So what you're seeing here is the conversion of those microRNA, those fibroblasts now turned into these cells that look like neurons. But so we've been spending a lot of time trying to understand this process. So going back to what these microRNAs try to do is the fact that they try to target a lot of components that are part of the chromatin regulation. And what we found was that when we topically express these microRNAs in fibroblasts, and if we measure the actual physical accessibility of chromatin accessibility by ATAC-seq, which measures the degree of chromatin accessibility by sequencing, what we found was that when human fibroblasts express these microRNAs, what happens is all these regions that used to be not so open, but they open up over time. And there are all these regions that used to be open and they close down over time. And this is a very controlled process, because if you look at all these regions that open up, they are usually associated with the chromatin regions that are nearby genes that regulate neural development, neural projections and differentiation and so forth. And another interesting aspect of this chromatin switching is the fact that all these regions that used to be open that close down, they usually co-localize with histone modification marks, including H3K27 acetylation and K4 methylation, which measures the which are the markers for basically enhancers of fibroblasts in this context, where you can see the closure of the enhancers that are associated with fibroblast context. So here are a couple of examples what they look like in terms of the sequence tracks. In upper levels, you're looking at ATAC signals where you see this gene, which is highly expressed in fibroblasts. And when we express these microRNAs, what you're seeing here is the closing down of all these regions around ECM1. And you can also see in the bottom tracks where these RNA-seq tracks where the transcript is actually being shut down. And here's another example, the chromatin region shutting off, closing down. And also there's a shutting off of RNA, the transcript for this gene called MFAT5, which is one of the markers of fibroblast. Here are a couple of examples of what the neuronal gene looks like in terms of chromatin accessibility control and transcript activation. So here you're looking at MAP2, which is one of the neuronal genes, the chromatin region opening up over time when they express microRNAs, and that usually comes with the activation of that gene over time. Another interesting aspect of is that if we look at other genes that are also expressed in neurons, but more specifically expressed in different neuronal subtypes, for example, MNX1, which is one of the markers of spinal cord motor neurons. What you're seeing here is that the region sort of opens up. There's no activation of that gene, meaning that there are also other neuronal genes, but they're more specifically markers of different neuronal subtypes that are being poised to be able to be activated. So with that, sort of gave us the rationale for how we combine these microRNAs with the different transcription factors that are expressed in different parts of the brain or distinctly in different types of neurons. And when we do this combinatorial approach where we combine microRNAs, let them induce the neuronal state, and at the same time, we combine them with different transcription factors, in this case, ISL1, LSX3, which are the transcription factors highly enriched in spinal cord motor neurons. Now we can push the conversion into spinal cord motor neurons. And here you're seeing the conversion example where now they activate HB9, which is the MNX1 gene, which is specifically expressed in spinal cord motor neurons, and they also become cholinergic neurons, as you can see from by the marker of chat. Okay. And the conversion work, this microRNA-based and also the transcription factor-based approach works pretty well in many different fibroblast samples from different individuals. So it's pretty robust system, and they also become electrophysiologically very active. So we try to understand what this process represents at more closer, more higher resolution in terms of the cellular dynamics. So what we found was that it is a very orchestrated processes of phage conversion. So what you're seeing here is a collection of cells that we collected at different time points of neuronal reprogramming, where fibroblasts now express MIR9 and MIR124. And we collected them at different time points, and we processed them through RNA-seq. And what you're seeing here is that the very distinct appearance of three distinct cell clusters. The cluster 1, it corresponds to starting fibroblast. And then there's a cluster 2, which is the sort of in the middle of conversion. And now the cells cluster into a distinct segregation where they're defined by the later time points, day 10, 15, 20. And the reason why we see this distinct clusters is because here's also RNA velocity analysis, which measures the sort of cellular state along the process of differentiation. And the reason why we see them as very distinct clusters is the fact that the first thing that microRNAs go in, and what they try to do is they try to recognize the non-neuronal characteristics in non-neuronal context. So here this is a plot called co-expression gene network analysis using Magena. So because we have a lot of N from fibroblasts, now we can try to build a network where that sort of genes that define fibroblast fate. And in the first time point where cells are pulled into a distinct cluster is represented by the fact that if you look at this network of fibroblasts and you express microRNAs, and you basically see the whole network goes away. And so we try to see what's going on with these microRNAs and how is that possible that the network of fibroblasts goes away. So we try to employ this method called AGO hits clip, which we pull down Argonaut and then we sequence the microRNAs and also the transcripts that are loaded into Argonaut. And we try to measure what are the targets that happen to be repressed by these microRNAs. So if we do that, and when we identify the targets of these microRNAs, what we are seeing here is the fact that microRNAs go in and they repress, or they try to abolish the fibroblast network, and a lot of components of this network is embedded with the target genes of these microRNAs. And another interesting aspect of this process is the fact that when microRNAs instruct this starting fibroblasts to lose their identity, so these are a couple of examples of fibroblast markers, and only after that, then the expression or the activation of the neuronal program kicks in. And this is seen by this cluster where now they start expressing the genes that are markers of neurons, including MAP2 and neurofilament and SNAP25 and whatnot. And here's another way of looking at it. It's a pseudo-time analysis where you see all these markers of fibroblasts the first thing that happens is these fibroblast genes, they get shut down, and over time, there's a stepwise activation of neuronal genes that occurs after the deletion or after the aggregation of the fibroblast fate. So just very briefly, that sort of represents our current understanding of neuronal reprogramming by this microRNAs. The first thing that happens is the fibroblasts, they lose their identity, and we actually identify the critical component of the process that microRNAs target the KLF family transcription factors called KLF4 and 5, and this is very important for losing the non-neuronal characteristics. And also there's upregulation of another small RNA called nuclear RNA called RN7SK, which is important for synergizing with the chromatin remodeling complexes to open up and activate neuronal genes. And during that process, that's where the transcription factors come in and to synergize with the microRNA-induced neuronal state to push them towards very distinct neuronal subtypes. So with this, using this paradigm, using microRNAs and also different transcription factors, then that's how we control what kind of neurons that we can generate. So for example, if we combine these microRNAs with these transcription factors called the CTIP2, DLX12, MIL1L, these are transcription factors enriched in the striatum of the brain. Within the striatum, there's a medium spiny neurons that are important for modeling Huntington's disease. Meanwhile, if we combine these microRNAs with ISL1, LSX3, we can push them towards motor neurons. And there's also other family of transcription factors called the NeuroD2, ACL1 and MI1L, that we can push them towards cortical lineage and also serotonergic neuron lineage. So the reason why we care a lot about what kind of neurons that we can generate with a high efficiency is because we're trying to use those neurons that are relevant for disease context. So I'll go into modeling purpose, where we try to model Huntington's disease, which is a neurodegenerative disorder. It is an inherited disorder caused by mutation in the gene called Huntington, and it's a polyglutamine expansion disorder, meaning that there is a CAG repeat number that when that exceeds the number of 36, and then that basically that defines the disease background, where when patients have more than 36 repeats of polyglutamine expansion, the disease kicks in around when people reach the middle age, and is characterized by massive death of striatal medium spiny neurons. From now, I'll just call them MSNs. And that, because of that, it leads to the movement disorder. It's 100% mortality after 10 to 15 years after the onset of disease, and there's no cure. So here you're looking at Huntington's disease patient brain, where this area is a striatum, and the majority of neurons that exist in the striatum is medium spiny neurons, MSNs, and because by the loss of the MSNs, and you see very massive degeneration of the striatum area. So as I briefly mentioned a couple of slides ago, we use microRNAs with CTIP2, DLX12, and MIL1L. We collectively call them CDM factors, and then we can basically turn human adult fibroblasts into medium spiny neurons. So we published these results, so I'm not getting into details. But one thing I really wanted to emphasize is the fact that the question of what is the age of these directly converted reprogrammed neurons. So our goal was to trying to see what is the cellular age that when we collect fibroblasts from human individuals, and how do we measure that, and what happens to that cellular age after they convert into neurons. So for this purpose, we mainly use this analysis called epigenetic clock analysis. This is based on DNA methylation. In our genome, we have about 350 loci where the methylation patterns are actually directly correlated to the actual chronological ages of human individuals. So what we did was we collected fibroblast samples ranging from baby all the way to 90 something years old, and we turned them into neurons. We measured the epigenetic clock and before and after the conversion, and the clock basically stays the same. And this is a universal clock that applies to many different cell types. So by this by the virtue of epigenetic clock analysis, we say the cellular age, or at least the epigenetic signature, is maintained during the microRNA-based neuronal reprogramming. And this becomes a very important aspect of modeling late onset disorders. So using this reprogramming approach, what we do is then we collect fibroblast samples from Huntington's disease patients, and we purposely focus on patient samples that have repeat numbers in the range of 40 to 50. So that defines when the adult onset population of HD patients, because that's the majority of the Huntington's disease patients. And the sort of inclusion of 40 to 50 repeats in mice is very hard to recapitulate the Huntington's disease associated pathway, the phenotypes. So our goal is to contribute to the field by being able to model this adult onset aspect of Huntington's disease progression. So what we do is then we take skin cells from the patients. They have the mutations because it's inherited, and we turn them into medium spiny neurons. And this is basically what the cells look like after neuronal conversion. This is the medium HD fibroblasts now turned into medium spiny neurons. So we collectively just call them HDMSNs. And the interesting aspect of about HDMSNs is that they recapitulate some of the adult onset HD pathology. And one of this includes the formation of aggregation of this mutant protein, mutant HTT protein. And this is very hard to see, but it's something that's seen in patient brains. So here you're looking at the examples of Huntington's disease fibroblasts. This is an example of medium spiny neurons turned from control fibroblasts. Now, this is an example of Huntington's disease fibroblasts now turned into medium spiny neurons. And as you can see, now they start expressing this or having this formation of HTT aggregation. And another phenotype is the actual neurodegeneration that occurs when they're turned into medium spiny neurons, including increased DNA damage measured by different ways. Here you're looking at accumulation of oxidative DNA stress, the DNA damage, and also double-stranded DNA break measured by 53BP1 expression. And the most, for us, the most important phenotype that we try to track in the lab is the actual degeneration of medium spiny neurons. And this is something that happens as a result of cells taking up the medium spiny neuron identity, because when we track them over time, there's not much difference during the process or fibroblasts of Huntington's disease patient samples versus wild type, but it's only after when they acquire the neuronal identity, and that's when they start degenerating. So we actually see degeneration happening in the tissue culture dish about day 35 into reprogramming. And another aspect of the modeling disease is the direct conversion of fibroblasts into neurons, but the phenotype is manifested differentially based on when we take patient samples. So for example, if we take patient samples before the onset of clinical symptoms, culture them out and turn them into neurons, and if we collect fibroblast samples from symptomatic patients and turn them into medium spiny neurons, So that generates two different population of medium spine neurons, all containing mutations. But what's important is, depending on what phenotype we look at, they have different manifestation of degeneration phenotypes. So what I mean by that is, for example, if we look at medium spine neurons derived from pre-onset HD patients, we still see the aggregation phenotype. But if we look at neurodegeneration, including the actual cell death and also DNA damage, is more pronounced in medium spine neurons derived from post-onset HD patients. So that really defines a platform where we can try to understand what are the age-associated changes that underlie the degeneration of medium spine neurons in Huntington's disease patients. So what we try to do in the lab is trying to compare medium spine neurons that we generate from pre-symptomatic patients versus symptomatic patients, trying to understand the differences between these two populations and see whether it's possible to make this symptomatic neurons to be more like pre-symptomatic neurons. So Youngmi Oh, and also with the help of Xiaowei Chen in the lab, what we are trying to do is we're trying to understand pathways that are defined, that are differentially manifested between pre-onset versus post-onset samples. And then what we do is then we take fibroblast samples from multiple patients, pre-clinical symptom and also symptomatic, turn them into neurons, and we basically try to compare these two populations in terms of what cellular state they are in. So all fibroblasts, they turn into medium spine neurons pretty well with a more or less equal efficiency. And then what we try to do is we try to, so we basically do RNA-seq and also ATAC-seq, and we try to understand the network that are more or less emphasized in different population of neurons. And so we do RNA-seq, and we basically do a lot of co-expression gene network analysis. In this case, it's based on WGCNA. And we try to detect the modules that are either lost or more pronounced in post-onset medium spiny neurons. So for now, we've after doing this type of co-expression analysis, what we do is we try to focus on one module. And for now, we focus on this module called the black module, because we're trying to find a network of genes that are lost in in symptomatic neurons. And meanwhile, we try to focus on modules that are less affected by the gender difference. And another interesting aspect of that is, if you look at the black modules and if you try to look at what are the potential upstream events that may be related to these differences in gene expression in black module. So a lot of genes that belong to this module have some or direct or some association with the autophagy pathway. And also the fact that if you predict the upstream regulators, they predict all these autophagy inhibitors, meaning that the analysis sees the symptomatic neurons to be more or less like sort of behaving as if the autophagy pathway has been inhibited. So with that, what we try to do is, if that's the case, then we can try to inhibit pre-onset medium spine neurons, and we see how if that treatment pushes the pre-onset medium spine neurons to behave more like post-onset neurons. And indeed, that's the case. So here you're looking at the measurement of neurodegeneration measured by caspase and also Anaxin-5. So by the way, now we measure the cell death with caspase activation, because we, at least in this direct conversion context, a lot of medium spine neurons go through the cell death by autophagy pathway found by Elias Turlin in the lab. So what Youngmi found was that if she treats pre-onset medium spiny neurons with this inhibitor called LY294-22002, which is the inhibitor of autophagy. And what she found was that now then pre-onset cells behave more like post-onset medium spiny neurons. And here's another example from an axon 5 measurement over time, where the pre-onset medium spiny neurons and they get more, they basically transition into more post-onset state by treating these cells with LY294-002. So this is some of the examples of how we use these reprogrammed neurons to understand how the different age state contributes to the onset of neurodegeneration. And the ultimate goal is to find pathways that are more pronounced or lost in symptomatic patient neurons, and see whether is it ever possible to push this symptomatic neurons to be more like pre-symptomatic neurons, where the cells are more resilient to neurodegeneration. So let me summarize by saying that we use microRNAs, miR9, 124, as potent neurogenic molecules that allows direct fate conversion of fibroblasts, human fibroblasts. And this occurs through stepwise processes where cells lose the fibroblast identity and they activate neuronal programs. And this microRNA-induced neuronal state can be synergized with other subtype-defining transcription factors to push the conversion into very distinct neuronal subtypes. And by doing so, we try to model different neuronal disorders by using patient-derived neurons. And because of the age information is transferred over to the reprogrammed neurons, we can now use these neurons to identify how the neuronal aging contributes to the onset of neurodegeneration. So with that, let me thank my lab members. A lot of the effort understanding how microRNAs initiate the reprogramming process is basically the whole lab's work. But the paper that I just described today about the stepwise process has been mainly pioneered by Kitra Cates and also Jisun Kwan, and also Matt McCoy, who is now doing a postdoc in a different institution. I'd also like to thank my collaborators within WashU and also outside of WashU and my funding sources. And I thank you for your attention.
Thank you, Andrew. We can take a couple questions for Andrew. Any speakers?
Hi, yeah. Hi, Andrew. Really nice talk, and then bring a different perspective for like a modern human disease. And I have two questions. So could the miRNA approach being used in the different cell type, and does the mechanism behind is conserved between the different species, like for example a mouse or some other organism?
Yeah. So so let me start with the different cell types. So the reason why we mainly focus on fibroblasts is because we try to model disease and fibroblast samples are readily available. But we have turned, like, for example, like smooth muscle cells into neurons, even HeLa cells into neurons, astrocytes into neurons. And so there's a commonality between different cell types that are non-neuronal. And I think what these microRNAs they tend to do is the recognition of this non-neural characteristics in non-neural cell types, and they repress their identity. So we haven't published that yet because it's something we worked on, and the sort of disease modeling aspect. So we kind of got busy with that approach, but it is actually doable to turn other cell types into neurons by using these microRNAs. So the second question is species. So initially I started this project using mouse fibroblasts, and after seeing it happening, so I basically transitioned into using human samples. So a lot of understanding came from what we did with the human fibroblasts, but I mean, like many years ago, I tried the mouse embryonic fibroblasts. Mouse embryonic tilted fibroblasts and also embryonic fibroblasts, and it works. It's just that we are not doing a lot of mouse embryonic fibroblast work. We have some samples from monkey fibroblast, and we actually haven't done a lot of work, but there's a group in Japan where they try to use marmoset fibroblasts and turn into neurons, and yeah. So I think there's some degree of species conservation.
Thank you.
There are two questions here. Are there other markers of fibroblast neuronal age that could be used to get a fuller picture of the age health of the converted fibroblasts?
Oh, that's an excellent question. Yeah, so it's actually one of the active research directions that my lab is trying to do. So, because we can't really measure epigenetic clock for every experiment we do. You know, that one figure that we showed, it cost us like $5,000. So we're trying to come up with a marker that are specific for different age groups. And when we do this perturbation experiment, and whether we can actually perturb the expression of the age-associated transcripts, it's an ongoing work. I don't have a full answer for it.
Okay. Great talk. If your patient fibroblast presents a mosaicism with some super expanded alleles, can your reprogramming generate an instability that would affect this mosaicism?
Yeah, so I think it's so now a lot of effort in the field has been focusing on genome instability, meaning that over time, these repeat numbers can expand. We only checked by collecting all neurons and run them through the sequencing to figure out how many repeats they still have after conversion. I think, so doing that, we didn't actually see strong evidence of the 40 repeats now being expanded. But I think when we did that work, the sensitivity may not be so primed to detect these subtle changes in repeat expansion. So I think this is more of an assay that needs to be done in single cell level. But we haven't been able to do that yet.
Thanks again, Andrew. Our next speaker is Martin Kampmann. He is an associate professor at the Institute for Neurodegenerative Diseases at UCSF. Welcome, Martin.
Thank you so much, Shasha, and all the organizers for putting together such a exciting, diverse program. Let me share my screen. All right, my lab is excited to use CRISPR-based functional genomics in order to uncover both mechanisms and potential therapeutic targets in brain disease. And Andrew's fantastic talk already introduced a lot of the themes. Brain diseases have been traditionally very difficult to study because of the lack of good human primary tissue that is available for experimental studies, and of course the limitations of certain animal models. But human genetics is giving us now a rapidly growing catalog of genetic variants that are associated with certain traits and diseases, including brain diseases. So that's exciting. But of course, then the next challenge is to really understand what the mechanism is, when and where these genetic variants might contribute to disease risk, and ultimately how we could translate such a mechanistic understanding into new therapeutic strategies. And this is a really important gap that we need to bridge. This graph here from a recent review by Alice Chen Plotkin's lab shows that in blue, the number of GWAS studies has been linearly increasing, steadily increasing over the last 10 or so years. Whereas the orange line here, it's not the x-axis, it's actually the number of functional studies that have been done on these GWAS hits. And you can see that we're really lagging way behind in our functional characterization. And so our lab, as other labs, is trying to basically use a functional genomics approach to bridge that gap. And by functional genomics, I mean the systematic, controlled and large-scale perturbation of genes in order to understand their roles in disease. And so I will briefly tell you about our functional genomics platform, and then specifically how we apply it to neurodegenerative diseases, although I think a lot of the lessons will apply to many other diseases that many of you might be interested in studying. When I started my lab a little over 5 years ago, we wanted to be able to do functional genomic screens in relevant cell types. CRISPR-I and CRISPR-A methods that we've heard about, that I was a little bit involved in co-developing as a postdoc in Jonathan Weissman's lab, had been mostly applied in cancer cells at that point, but we wanted to study brain disease in relevant cell types such as neurons and glia. And we decided to implement these technologies in iPSC-derived differentiated cell types because that would enable us to really work with patient-derived cells that have specific genetic variants. Raylin Tian, fantastic graduate student in the lab, led this effort and was able to generate iPSC lines that have a stably integrated CRISPR-I machinery. We now also have a CRISPR-A machinery and genome-wide screens with that on bioRxiv. But in this first CRISPR-I paper, he showed that he can very effectively knock down individual genes in differentiated neurons, here knocking down, for example, progranulin, which is associated with frontotemporal dementia. And not only, of course, studying one gene at a time in kind of a reverse genetic way, but to actually use this for massively parallel genetic screens by introducing guide RNA libraries into iPSCs and then creating a number of phenotypes that we can look at in these massively parallel screens, such as neuronal survival, giving us a set of essential genes, which are interestingly different from the types of essential genes people had detected in cancer cell lines. So that definitely neuron-specific essential genes, but also coupling gene perturbation to very high dimensional readouts, such as single cell RNA sequencing, where for each individual genetic perturbation, we would get a rich fingerprint of the cellular pathways that get up and down regulated in response to that perturbation. And also, of course, we can couple it to high content readouts and arrayed screens where we could look at phenotypes such as morphology, but also in unpublished work, neuronal activity, which is ultimately obviously one of the most interesting things that neurons do, using first kind of optical reporters of neuronal activity and then coupling it to more classical electrophysiology. So this is published work, so I just briefly highlight 2 vignettes before I tell you about unpublished work in which we apply this. And I just wanted to highlight these 2 vignettes because they really brought home to us that it is important to study the biology you're interested in the relevant cell types. These 2 heat maps basically summarize these experiments using the crop-seq method, where we perturb genes, and the genes we're perturbing, they're too small to read, but they're along the y-axis here. And then we're observing in this pooled experiment what the transcriptomic changes are that we observe. And along the x-axis, again, too small to read, are all the transcripts that change in response to the perturbation, either up in red or down in blue. And as you can see, this is not a random scrambled heat map, but there's a lot of structure in there. And we have iPSCs on the top and neurons on the bottom. You can, for example, see that there are clusters of stereotypically upregulated genes. For example, this red cluster here are genes involved in lipid biosynthesis, and knocking down any of these genes here that are involved themselves in lipid biosynthesis triggers upregulation of this pathway. So that would be a classical homeostatic response. And that response, for example, is somewhat conserved between neurons and iPSCs, but we also saw some really interesting differences. And I just will highlight one single gene that I thought was quite interesting to us. And this is a gene called UBA1. UBA1 encodes the ubiquitin activating enzyme. So if you're familiar with the cascade of ubiquitination in the cell, you have the E1, E2, E3 enzymes. And the E1 enzymes are just on the top of that cascade, activating ubiquitin for then transfer to different proteins. And so the UBA1 is the main E1 enzyme. So as one might imagine, it's really essential in all cells. It's a housekeeping gene, I would say. Knocking it down was toxic both in iPSCs and in neurons derived from these iPSCs. And so far, so good. That was not a big surprise. But then when we looked at the transcriptomic consequences of knocking it down, this is an MA plot. So on the y-axis, you can see log2-fold change, and on the x-axis, average expression of different transcripts. First of all, UBA1 gets very effectively knocked down, both in iPSCs and neurons, roughly expressed around similar levels. Knockdown works very well in both cell types. But when we now look at the other transcripts that change in response, the picture is very different from neurons, between neurons and iPSCs. So for neurons here on the bottom, we saw a number of genes being upregulated that are so-called heat shock proteins encoding molecular chaperones. And that's a signature that's well known to be activated in response to proteotoxic stress. So for example, you could imagine that without ubiquitination, the turnover of proteins, including misfolded proteins by the ubiquitin proteasome systems is impaired, and that would be predicted to trigger upregulation of these molecular chaperones. Interestingly, we see also other stress genes come up, including ApoE, which of course is a gene of great interest for Alzheimer's disease. In iPSCs, that signature is completely absent. We do not see this upregulation of heat shock proteins. Instead, we see a different, also coherent signature, downregulations of all of these so-called metallothioneins. That is a signature that had been previously linked to oxidative stress. So this was just very interesting and told us that, either UBA1 is lack of function triggers different responses in different cell types because they have evolved to respond differently to the same stress, or maybe more likely, UBA1 knockdown causes different types of stress in these two cell types, because maybe given that it is a pleiotropic gene, different aspects of its functions are limiting in these different cell types. And so this just really highlights the importance of studying a gene in the correct cell type. One more interesting thing about UBA1 is that partial loss of function mutations of UBA1 in humans are linked to neurological symptoms and neurological disease. Again, interestingly, even though UBA1 is expressed throughout the body, we think of it as a housekeeping gene. Specific point mutations have cell type and tissue-specific consequences also in human disease. And of course, pooled screens that we can couple to a readout such as survival, flow cytometry, and single-cell RNA sequencing are very nice and scalable. But we are also interested in high-content array screens where we would basically introduce different guide RNAs, one per well, in a multi-well format and read out phenotypes, for example, by high-content microscopy, because that allows us to access phenotypes that are disease relevant, but that are hard to evaluate in pooled screens, such as, high dimensional phenotypes, aspects of neuronal activity. Importantly, also non-cell autonomous phenotypes, interactions between neurons and glia, something I will come back to at the very end of my talk. So here's just an example of this kind of high content coupled to CRISPR perturbation. On the left, this is a time lapse of neurons developing over a couple of weeks and with a non-targeting control guide RNA. So this is what our wild type neurons would look like. And then on the right, we're knocking down one of the genes we had identified as an essential genes in neurons. It's actually an electron transport chain component. And you can see these neurons, a lot of them die. They fail to properly differentiate or develop axons. And maybe not surprising if you're knocking down an essential gene. So you could say, so what? Do we learn anything from looking at these sick neurons? Well, it turns out we have kind of an Anna Karenina situation here where all the happy neurons are the same, but all the sick neurons or all the unhappy neurons are unhappy in their own way. And Ray Lin developed a platform to basically segment the images we got from these neurons and extract metrics. For example, some very simple metrics such as how many neurites come off the cell body, what's the length and the branching degree. And doing that, he could extract a lot of information from these microscopy images. Of course, both about their survival rate, where we got a nice agreement between microscopy-based survival estimates and the pooled screen survival estimates, but also looking then at neurite outgrowth and some of these other morphological phenotypes and seeing how different guide RNAs that targeted the same gene had quite the same pattern, but then guide RNAs targeting different genes had their own idiosyncratic pattern. This is summarized again in a heat map here. Each row is a guide RNA, and each column is one of the metrics extracted by microscopy. And in white would be what non-targeting controls look like for each of these metrics, and then positive and negative deviations are shown in red and blue respectively. And so the cool thing, I think, was that when we did hierarchical clustering of these rows to ask which knockdowns have similar patterns by microscopy, even though literally mostly we're looking at dying neurons, there was so much specificity that for quite a few of the genes, the two guide RNAs targeting that gene, independent wells, independent reagents clustered right next to each other. So there was enough specificity in looking at these sick neurons to say, this neuron has a problem with cholesterol biogenesis, this one with electron transport chain complex 5, this one with endolysosomal trafficking. And that was very exciting and actually reminded me of work that has been done in machine learning, where, for example, people were able to just look at children's faces and predict which genetic disease they had. So ultimately, we're very interested to get these high-dimensional phenotypes. We established, together with Data Technica International, a new data commons called CRISPR Brain, where we're basically having both low and high dimensional phenotypes from CRISPR perturbation screens for different cell types. We already have 250,000 different phenotypes in there for cell types, but we hope that this will expand rapidly because the iPSC technology uniquely allows us to really compare side-by-side phenotypes in different human differentiated cell types. This is a community data commons. We hope that many of you who are engaged in CRISPR screens in different cell types are interested in contributing your data. We'd love to feature it and help you upload it. So any feedback is very welcome, and you can reach out to me if you'd like to engage with us on this. Ultimately, the vision is that we can get a lot of high-dimensional, interesting phenotype that includes morphology, as I showed, but also, as I mentioned, neuronal activity, transcriptomics, and other cellular readouts, and that we can ultimately integrate them in order to really understand how, for example, a specific disease gene affects all of these phenotypes, and also how they map to each other. Is there something in a transcriptomic signature that would correspond to a change in neuronal activity? That would be very exciting because we are now generating more and more of these transcriptomic data sets from human postmortem brains, but we can't do functional studies in them. So if we crack that code of what features in the transcriptomics correspond exactly to which functional readouts, that would be great. And furthermore, our CRISPR perturbation platform allows us to kind of create a lookup table of what perturbation has which consequences in these different cell types. But now I want to talk about unpublished work and specific applications to neurodegenerative diseases. The CRISPR-I and A platforms that allow us to really change expression of endogenous genes up and down has two really important classes of applications, I think, to better understand human genetics. One of them is that a lot of the human genetic changes are non-coding and are likely to act by expression, by affecting expression of other genes. Sometimes these are nearby genes, but not all of them are nearby. So these so-called EQTL might also have cell type specific effects on gene expression. And then it's not so easy to understand, what do they actually act through? So one of the things we can of course do is that by using CRISPR-I and A, we can specifically model the changes in candidate genes that might change as a result of an EQTL genetic variant in different cell types, and then evaluate the functional consequences, including for very high dimensional phenotypes such as single cell RNA-seq, and then to even see whether different EQTLs linked to a similar disease risk, for example, have some convergent phenotypes to really help us see whether there are convergent mechanisms on the cellular level in specific cell type. Today, I will talk more about the other application that we can use CRISPR-IA screening for. And those are modifier screens that help us understand disease mechanisms and, importantly, to also uncover potential therapeutic targets. As I mentioned, we were very motivated to work in iPSC-derived cells because we can derive them from patients, for example, who have a specific disease-linked variant. If that variant is known, we can do CRISPR editing on the iPSCs to create isogenic controls. We can then introduce our CRISPR machinery and guide RNAs, differentiate them into cell types that we believe are relevant for the disease that we want to study, and then detect whether the cells carrying the disease variant have cellular differences, cellular defects maybe, that mirror what happens in a patient brain compared to isogenic wild-type control. Many people have done that. We've heard some nice example from Andrew for Huntington's disease, and people have observed these differences. But importantly, in most cases, it's not really understood how you go from a single base pair change in the genome to a complex cellular phenotype. So we can now do modifier screens, genome-wide modifier screens using our CRISPR-IA method in these cells to try to understand mechanisms and therapeutic targets. So once we have identified this phenotype of interest that we think is disease relevant in that cell type, we can ask for all the genes in the genome when we change their expression level, do they make that phenotype better or worse? And of course, we can do that in parallel in the control background and the disease background. As you can imagine, for any phenotype you pick, you might find modifiers that are modifiers regardless of background, right? And so that might tell you about that phenotype, but maybe less about the disease. But those genetic modifiers that specifically affected in the disease background, not the healthy background, you could think of them as genetically interacting with the gene variant, can give us mechanistic clues. And of course, a subset of them are potential therapeutic targets that might improve the cellular health or the phenotype of interest, even in the presence of the disease mutations. Now, so I think while of course the long-term vision for many of us will be to do gene therapy and actually correct a genetic variant, this is obviously really challenging in the context of brain diseases still. And so if instead we find some other potential targets in other pathways that are maybe druggable with small molecules, this could be a great, very actionable way to have new therapeutic options for these devastating diseases. So I'll show you one specific example in unpublished work from our lab where we're applying this paradigm. And one of the cellular phenotypes that has been known as a hallmark of neurodegeneration for more than 100 years is protein aggregation. The fact that specific proteins aggregate in specific cell types at a certain age in these diseases. And this is a rogues gallery of some of those proteins that aggregate in different diseases. And importantly, the big question to ask, of course, is this protein aggregation causal in the disease, or is it just incidental? And I think the best evidence that there's something about these proteins and what they do is causal is the fact that for any of these diseases, there are rare familial cases that are actually linked to point mutations in the protein that aggregates, or overexpression or changes in the way it's processed, and other things like that. In Huntington, as we heard, linked to polyQ expansion. So these diseases are something about those proteins can cause the disease in a 100% penetrant way. And so whether it's the large aggregates that cause the disease or something on the path remains to be determined. And we focus a lot on a protein called tau, which aggregates in Alzheimer's disease and related dementias. And Avi Samerson, a postdoc in the lab, investigated iPSCs we received from Li Gan's lab that had a mutation in tau, gene name is MAPT, V337M, that gives people frontotemporal dementia versus isogenic wild-type controls. And when Avi differentiated these neurons, already at 6 weeks of differentiation, he saw by Western blot that the mutant but not wild-type neurons accumulated a phosphoform of tau, recognized by this 88 antibody that is a hallmark of Alzheimer's disease. So neuropathologists will use that same antibodies in brains. And maybe more excitingly for us even, using a conformation-specific antibody that recognizes tau oligomers. So what we think of really early stage changes before you even form big aggregates. At 2 weeks of differentiation, already they were oligomers detectable in the cell body of V337M neurons, but not wild-type controls. And Avi established a way to do flow cytometry on these fixed-state neurons and found that V337M neurons again are shifted towards higher levels of tau oligomers by flow cytometry. So with that assay in hand, Avi was able to do a genome-wide modifier screens introducing a guide RNA library that targeted all 20,000 protein coding genes into these iPSCs, differentiated them into cortical glutamatergic neurons, stained them with this tau oligomer antibody, and then this FACS sorting to physically separate those neurons that had high levels of tau oligomers versus low levels of tau oligomers. And then on these two bins, did targeted next generation sequencing to count frequencies of guide RNAs in both populations. So the concept being that if you have a guide RNA that triggers higher levels of aggregation, cells expressing that guide RNA would be more frequent in that bin, and vice versa for guide RNAs lowering aggregation. These are the results from the genome-wide screen shown in the so-called volcano plot, where on the x-axis you have things that either increase tau oligomers on the right or decrease tau oligomers on the left when knocked down. And here we see the statistical significance. And so first off the bat, there are a lot of significant hits. One of the top ones is knockdown of MAPT, tau itself. This is a nice technical control, right? Knocking down tau, you should have fewer tau oligomers. It also is a reminder that this screen was not designed to exclude hits that would act on total tau levels. And we did that on purpose because there's a lot of interest in just lowering tau. It's being pursued by antisense oligos therapies in clinical trial right now, because the thought is that really in disease, tau aggregation is about a gain of toxic function, and even just lowering total tau is beneficial. And we know in secondary screens actually deconvoluting which of our hits acted on total tau levels versus aggregation of tau. And we have both types of hits actually. TDP43 was interesting to see. It's a disease gene actually classically linked to ALS and other forms of frontotemporal dementia that do not include tau. But intriguingly, pathologists have very recently described that TDP43 can be an additional risk factor in tau in the context of Alzheimer's disease and tau aggregation there. So it's intriguing to see whether we might have a cell-based model to understand that interaction. We found a lot of factors that are important for the early stages of autophagy. And it's been previously described that autophagy is a clearance mechanism for tau aggregates. So it was nice to see this very coherent signature here versus mTOR, which is a negative regulator of autophagy, knocking down mTOR was actually beneficial and lowered tau oligomers. And this is actually something that had been described in a recent paper that did a small molecule screen in iPSC neuron models of tauopathy that also described that mTOR inhibition to induce autophagy was beneficial. So it's nice that our genetic screen can also come to the same conclusion about what potential therapeutic targets are. When we did an unbiased GO-term analysis of our hits, the top category of genes was stress response, so that includes things like molecular chaperones, which we're now studying to understand whether they directly interact with tau and what they do in that context. Maybe to our surprise, the next 3 GO-terms down the list all had to do with mitochondrial function. And so when we look at where these hits are, you can see that almost all of them, when knocked down, increased tau oligomers. So that was intriguing. And we could recapitulate that by inhibiting the same pathways, such as complex 1 with rotenone or mitochondrial protein synthesis with doxycycline, and recapitulated this increase in tau oligomers. And we're now following up mechanistically what that means. One hypothesis is that this causes reactive oxygen species that might directly oxidize tau. We're looking at that, but there are obviously many other indirect mechanisms. We are excited about this because, of course, one thing that is known about aging neurons is that they start to have mitochondrial defects. And so it is possible that we're here uncovering one of the reasons that tau aggregates in an age-dependent manner, because some of these knockdowns might model some of the defects that neurons have with aging. And there are many other hits that we're exploring that are potential therapeutic targets because they lowered tau aggregation levels. This was looking at tau aggregates. Of course, the other question is, why are tau aggregates actually bad for cells? And people don't really have that answer. Greg, a student in the lab, is looking at some paradigms for that. For example, he was able to reproduce work from other groups that had shown that low concentrations of rotenone that are not actually at all toxic to wild-type neurons are selectively killing V337M neurons to a greater degree, and knockdown of tau rescues that. That was an important control, but also knocking down some of the other hit genes from RV screens, including genes that do not lower total tau levels, but just block its aggregation where beneficial. So that was exciting to see because it really suggests that something in the process of tau aggregation is upstream of toxicity, and the tau aggregation screen could identify factors that block aggregation, but Greg is now doing a screen also just based on toxicity to maybe identify factors that block toxicity downstream of aggregation. Because if you think of disease progression as this arrow, of course, we'd love to have therapeutic targets that can intervene slightly later in disease when tau has already started to aggregate. Just really briefly to mention that this was up to this point a neuron-centric talk, but if we look at Alzheimer's disease risk genes, for example, where they're expressed, yes, they're expressed to some degrees in neurons, but other cell types, in particular microglia, are really enriched for them. And other studies have in fact pinpointed the fact, as, for example, recent work by Chris Glass, that even the specific non-coding variants can be in microglia specific enhancers of genes that are then linked to Alzheimer's disease. So we are also establishing our method in other cell types. Nina and Sid in the lab have a great system up and running for CRISPR-I and CRISPR-A and iPSC-derived microglia cells where they can very effectively knock down genes. Here showing the Alzheimer's risk gene TREM2. And Emmy in the lab is starting to put it all together and constructing 3D assemblers of neurons, astrocytes, and microglia that enable her to really look at cell-cell interactions and also dissect which cell types a specific disease gene might be acting through. So with that, I just want to thank our lab. I mentioned lab members throughout the talk who contributed to this work. The technology development was in collaboration with Michael Ward, and Lee Gann contributed the V337M neurons. These are our funding sources. And yeah, thank you for listening. I'm looking forward to your questions.
Thanks, Martin. That looks like a fun trip.
Yes, this was pre-COVID, but hopefully we'll all be together soon again.
So I have one question here. I recall reading that targeting A-beta in Alzheimer's mouse models showed limited to no non-therapeutic benefits? What would be some reasons for this, given that A beta aggregates are so associated with AD?
Yeah, this is obviously a really long-standing question in the Alzheimer's field, right? As you may know, there's been a decade-long controversy, whereas, for example, A beta is the more important hallmark and driver of disease versus tau, for example. A beta is, has received a lot of attention because mutations in the precursor protein APP and the presenilins are linked to early onset Alzheimer's disease. So it can set off a cascade. But when you actually look in the brain of patients locally, for example, tau aggregates are much more correlated with neuronal defects and then neuronal deaths than A beta. We do think A beta is important, but tau might be more proximal to actual neuronal dysfunction. So I'm really excited, why clinical trial phase is always a really complex question. It could happen for many reasons. I don't think it disproves that A beta can be important in disease, but with Alzheimer's, it's also really challenging to understand when you need to intervene therapeutically. But tau seems to be downstream of A beta possibly, and so intervening with tau might be a way to block disease progression, not only in Alzheimer's disease, but also in tauopathies that do not involve A beta, such as traumatic brain injury in soldiers or football players where tau aggregates, but it's less linked to A beta.
Hi, Martin. Really nice talk. So I have two questions. So you touch base on the cell non-autonomous effect for the disease, and then also try to use the 3D model. So I just wondering, because you nicely show you can do the phenotypic screening in the 2D model. So I don't know what's your approach to, if you have mixed population of cell type, and what's the approach you do dealing with the 3D model system?
Great question. One of the reasons we established this 3D assembloid model is because it really enables us to only have, for example, CRISPR machinery or perturbation specifically in one cell type, but then also look at effects on other cell types. It also enables us, for example, to mix and match isogenic cell types with genetic variants. The one example that's one we're working on is ApoE, which is a gene that has common variants in the population, ApoE 2, 3, and 4, where 4 is linked with extremely high risk of Alzheimer's disease. And it's not clear which cell type ApoE4 is working through. It's also secreted. So basically, there's two layers of cell type questions that we have. And so the assembloid models enables us to mix and match ApoE alleles in the different cell types, neurons, microglia, and astrocytes, to investigate effects on other cell types. And then we can go in and do a modifier screen. So for example, we could ask, what are genes that control survival of a neuron in an ApoE4 glial environment? So that's one example.
Luke has a question.
Yeah, hi, Martin, great talk. I had a question sort of just on functional genomics methodology. Are there key considerations you think about in screening between proliferative models and non-proliferative models? Or can you comment at all on that? I mean, a lot of work has gone into cancer models, but this is a new space in these post-mitotic cells.
That's an awesome question, Luke, and something we definitely were maybe concerned about initially. The two key things that come to mind that are easier maybe in proliferative models is if you are looking at cell growth defects. In a proliferative model, you could do the screen over several cell divisions, and even small differences would be amplified at each division, right? Versus in a non-proliferative cell types like neurons, there's only one shot to look at modifiers of survival, basically. And furthermore, thinking of now CRISPR perturbation, of course, if you perturb a gene by either knockout or knockdown in a dividing cell type, the gene product will be diluted out to cell division, versus in a post-mitotic cell type, if the protein is long-lived, you might not see that effect very soon. So I do think that in principle, that means that these screens might be slightly less sensitive than in dividing cell types, but we were very happy to see that we will still be able to pick up a lot of modifiers, even of, say, survival in post-mitotic cells. I would say an ongoing challenge is to have a good time resolution of an inducible system and a good degree of knockdown in post-mitotic cells. And that's something where we are trying to improve inducibility in those types of systems.
There's one more question for you, Martin, from Bruno Benetes. Lovely talk, Martin. In your genetic screening on tau aggregation, did you find any GWAS hit for AD or any other tauopathy?
Yes, that is absolutely something we're looking for. And I would say TDP43 is an interesting one that's kind of emerging as a modifier also of tauopathies and Alzheimer's disease. And that's why I showed it. But down the list, there were a few weaker hits that we still have to validate that also fell into those categories. Having said that, interestingly, Alzheimer's disease, a lot of the GWAS hits seem to really be acting in microglia. So I think those hits we'll probably uncover more once we start to do really larger scale screens in these co-culture models.
Oh, thanks again. We are going to our last talk by Michael Kosicki. I believe he did the work in Alan Bradley's lab as a graduate student at Wellcome Sanger Institute. And now he's at Lawrence Berkeley National Lab. Michael's title is Large Deletions and Small Trouble.
We can't hear you.
Okay, well, now you should.
Now we do.
All right, perfect. Yeah, so again, thank you for organizing and inviting me, and welcome to the last of the day. I hope you will learn a little bit about the more traditional uses of CRISPR-Cas9, which involve double-stranded breaks, and you would learn what to watch out for, and hopefully a little bit about the DNA damage repair in the process. So we all know how it works. Luke actually scooped me a little bit on the introduction, so I'll be brief. Cas9 introduces double-stranded breaks as its target site, and what you normally expect to see as a result is a bunch of indels, mostly small deletions of a few base pairs and sometimes insertions of one or two. And because it's so efficient and because it happens pretty often, few people early on have asked the question of what else can happen and, is it relevant to our experiments? And this is the essence of the project that I done during my graduate years in Alan Bradley's lab. I was trying to understand what other events happen and to select for them and describe them in detail. And this is what I did by instituting this little selection system, working with mostly mouse embryonic stem cells, which have the advantage of having a defined ploidy, so 2 copies for autosomal genes, and those particular cells that I use were male, so there's only 1 X chromosome in them, as opposed to, say, 3 or 4 in some cancer cell lines that people have been working with. And okay, I take those cells, I transfectome them with guide RNAs against different targets. The system I've used was very saturating, so all the components were integrated so as to achieve pretty much 100% cutting and mutagenesis. And after all is said and done, you would expect that the target site to be, there to be multiple different mutations that I generally divide into small indels and large deletions. And if you choose your target right, for example, if you target the intron of a gene, then the small indels would not have a phenotypic effect usually, whereas the large deletions would influence the expression of the gene in question. And that allows you to assess the frequency of those events by flow cytometry, for example, and you can also select the events that have led to loss of gene expression and analyze them in more detail, see what the actual underlying genotype is. And again, this is helped by using a target on chromosome X in male cells where there's only 1 allele, and those events stand out very clearly rather than having to reach sort of a homozygous event on other chromosomes. So that's what I did, and yeah, you've seen this particular slide before. So this is the result of selection for the events that are affecting the gene expression in the intronic cut. Realized here is a vertical line, and each line is a single cell cloned allele, or essentially a result of sequencing a single cell cloned cell. And you can see that most of the events are indeed large deletions, although there is also a couple of others that are very interesting. For example, there is those non-contiguous lesions where there is an indel on at the cut site, but also a small deletion or indel near the cut site. So this is also something that you have to watch out for. There is also large insertions, mostly from the plasmid or sometimes even from the genomic DNA of E. coli that sometimes is part of your plasmid prep. So quite a lot of different outcomes. And one thing that I haven't been highlighting before, even really in the paper, is that we've had a couple of clones with unresolved outcomes that after a few sort of genotyping steps, we've managed to figure out, well, would be unresolved deletions of even larger extent or translocations. So there's quite a bit of different outcomes you can expect. And there is also, well, obviously some groups that have been looking at those in different organisms before. I just want to sort of give a shout out to them in here. One big question is obviously how big of a problem is it? How often does it happen? And I think our best estimate looking at this experiment and also others that we've done and that other people have done since then is that large deletions seem to occur at around 20% per allele frequency. And this is fully consistent with the fact that if you edit a regular autosomal locus and get two of these events at the same time, that would only constitute 4% of your say single cell clones, a level which most experimentalists would consider acceptable as sort of a loss of DNA or a mishandling of cells, or something happened that we don't really care about, which is probably why these events haven't been quite appreciated before. On the other hand, obviously in say a clinical setting, 4% of cells with large unpredictable outcome could be a problem. So we still think it's quite important to take that into account. In any case, all this work has been already published, and what I thought was really interesting in the meantime, a few other things as well happened, and this is just, again, sort of listing a number of different papers that have come out that sort of were relating to similar problems. Especially there's a whole score of papers looking at loss of heterozygosity that can be the effect of Cas9 cutting or loss of parts of chromosomes as well. And it seems to, well, the loss of heterozygosity seems to have first been reported by Matthew Porteous, but then there's been a very nice score of papers from Dalpov, Nayakan, and Egli labs, and I do recommend especially the Egli paper that has officially come out now, which is a really, really beautiful study of effects of Cas9 in human embryos. Yeah, there's been also very nice thorough work by Charlie, who has spoken yesterday, Charlie Gersbach, who talked about the effects in context of gene therapy with AAV vectors, and also very much focused on the transcriptomic changes, which is really interesting. And yeah, there's a bunch of other papers that I won't go in through in detail, but that definitely confirm that there's many different aspects of on-target effects, of on-target unexpected outcomes at the cut site of a Cas9. With all this in mind, I think one thing that has been relatively unexplored and sort of an obvious question going forward was what particular mechanism leads to these outcomes? What can we learn by looking at the DNA damage repair? And this is a sort of simplified cartoon of what double-stranded break repair looks like in particular. And one way of thinking about it is as sort of progressing through different levels of resection. So if a cell can deal, so to speak, with a double-stranded break immediately by simply religating it, or maybe processing it in a very limited fashion, especially if it's not cycling and the sister chromatid is not available as a template, then it would go through non-homologous end joining, which usually results in all these small indels, or at least so we understand it. But then a sort of very important branching point in this pathways is the MRN resection, the MR11, RAD50, and NIBIN resection, which allows the cell to either go further, resect further, and end up with homologous recombination that requires those resected ends for invasion of the sister chromatid and templated repair, which usually should be perfect. Or if that fails or is not available, or time run out and the cell cycle must move on, then often what happens is some sort of microhomology mediated end joining, or possibly a single-stranded aligning outcome, where microhomology mediated end joining is of particular interest in here because it has been reported to involve largish deletions, maybe not necessarily the size that I've been talking about just now, but in general, these sort of events. And that's particularly characterized by pol Q gene or pol theta protein, a protein with many different functions. In any case, I decided to sort of take stock of all that and try and make a screen, essentially, to understand these things. Oh yeah, one more little detail is that when homologous recombination ends up perfectly, obviously we end up in the same place sort of as we started, and Cas9 can keep on cutting. So that was sort of the general expectation, that homologous recombination would not be an important determinant of large deletions, but yeah, that just as an aside. In any case, I've created this little library of Cas9 positive mouse embryonic stem cells that were deficient for many double-stranded DNA break repair genes, and it's all stems from one single Cas9 positive parent. So as opposed to perhaps a strategy which would involve me sending a lot of emails to different people and sourcing their particular cell lines, this is very well controlled. In fact, I of all the almost 100 clones that I had, 12 of them are control clones transfected by different non-targeting guides, and of the clones that were deficient or bona fide deficient for different DNA damage repair genes, there's 32 genes targeted in there, and the knockouts were verified by the sequencing of the targets of guides that I used to knock out the genes. So this is not a perfect way of assuring loss of actual gene function. It only tells you that there is no in-frame alleles or frame-preserving alleles, but as we know, cells can find a way to circumvent those things by alternative splicing and alternative start sites. So not all of these clones will necessarily be knocked out. In any case, I took this library and transfected it with guide RNAs, repeating the sort of experiments of the past. And I looked at the results in two different ways. One of them was to look at the sheer frequency of large deletions, and here defined essentially by the distance from the nearest exon, which was roughly 250 base pairs in the case of the guide that I used. And also I have sequenced the small indels by targeted sequencing. So essentially generating those indel profiles where negative numbers here means deletions of a given size, and minus 30 collects everything larger than 30 base pairs. And on the other hand, the insertions. Right. So what are the results? Well, first of all, we get the usual suspects for the large deletion frequency. So controls are in here, slightly variable. We'll come back to that. But what really stands out is that almost any part of the core non-homologous end joining pathway you knock out, and that includes Q7080 that are here called XRC5 and 6, DNA PKCS, XLF, XRC4, LIG4, all the sort of big players dramatically increase the frequency of large deletions. And I think this is particularly important considering that many strategies for increasing homologous recombination repair using a template involve essentially knocking out or inhibiting this particular pathway. So be careful what you wish for, I suppose. And then there is the second group of hits, which are, well, essentially two proteins in my particular screen. POLQ, so Pol theta, that I mentioned, is sort of the core microhomology-mediated end joining protein, and also Nibin, which I also mentioned is a core resection protein. And it is well known that the other elements of the resection complex, the MR11 and RAD50, are pretty much impossible to knock out in full because that's lethal, but without Nibin you can obtain viable cells that are severely impaired for resection. So far, so good. One very interesting observation I thought was that because we both measured the frequency of large deletions and the small indel sizes and shapes, so to speak, we can actually compare the two, and we find a pretty strong correlation between the frequency of large deletions and the size of the small indels. And very specifically, the larger the more frequent the large deletions, the larger the small indels. And this implies very strongly to me that there is no specialized large deletion pathway, that it's simply a continuum of resection and repair, which determines the size of the deletions and their frequency. Right. Having said that, I decided to investigate the indel profiles in more detail, and I was quite lucky to have worked on targets that in wild-type cells result in very different indel profiles. So we have this one target which results in pretty much exclusively +1 insertions, and then there's this one which results in a more typical or average repair profile with small deletions, the primary outcome. And then we have this really funky one with sort of very strong attractors at -2, 10, 20, and sort of larger deletions. So whole gamut of different outcomes in wild-type cells. And then we can obviously compare those outcomes to outcomes in cells with particular knockouts, and we'll see that the knockouts in non-homologous end joining pathways generally sort of push, if you wish, those profiles towards the left, towards larger deletions, as consistent with the actual large deletion screen, whereas knocking out, say, POLQ pushes it to the right, so to non-resection outcomes, mostly small insertions. And of course, I could look at all the profiles and show them to you for the next hour, but that would be a little bit dull. So I was looking for a method to organize all this data and simplify it. And I've used sort of a concept from a colleague of mine at Sanger Institute who are using the KL divergence as a measure of difference in between different indel profiles. I computed that for every pair of samples I had for all the different knockouts and controls, and then deconstructed that through a method very similar in form to PCA, which is called multidimensional scaling, effectively ending up with something that looks like a PCA plot. And as you will see, most of the clones, as perhaps expected, most of the knockouts don't do anything, and they cluster together in the middle with the control clones. Whereas the usual suspects, the really strong players in non-homologous end joining pathway and the POLQ and Nibin, are at the edges and also at the sort of polar opposites. So everything seems to check out nicely in this case. And then we can obviously look at those profiles for all the 3 targets that I've looked at. And we actually can see, well, both the commonalities, obviously, the sort of usual suspects are where you expect them in every single one of the 3 guides. But then you also can see some targets that seem to be very specific to particular guides. For example, POL-L in here, or Artemis, DCLRE1C, and DNA PKCS in here, and others as well. And also T-Rex1 in this particular case. So what makes those particular targets sort of different, we can see that by sort of examining the plots that are sort of differential plots between the knockout and the wild type. So for example, for pol2, knockout with this guide, it definitely seems to affect the strongest outcome, so the +1 insertion, which makes sense knowing that polymerase lambda pol-L is an important protein that adds single nucleotides at double-stranded breaks. So that makes sense. Yeah, there is also DCRNE, so the Artemis, which is a little bit more difficult to understand. But at the end, we have TRX1, which was a fascinating hit because I essentially included it just because it's an exonuclease, but not because of any prior evidence that it should be involved in double-stranded break. In fact, it is well known to be involved in autoimmunity through controlling or degrading single-stranded DNA in the cytosol of the cell, which shouldn't, in principle, have much to do with double-stranded break repair. However, in this particular screen, it's clearly has a very specific role for a specific indel, -20, and actually it also has a pretty strong role for one of the two major -10 indels, which are here aggregated together. So yeah, we are really excited about this particular thing, and I would just want to mention one name, John Maciejowski, who has been doing a lot of interesting work, well, by himself now, but as a group leader, but also with Peter Campbell and Ticia de Lange on role of TRX1 in destroying chromatin bridges or resolving chromatin bridges, which implies that the protein itself is at least present around either in the nucleus or, as he recently found, in the micronuclei related to DNA damage repair, which maybe suggests how it works. But it's an interesting hit in any case. Finally, I would like to stress the sort of one direction that I was always a bit interested in as to how the intensity of mutagenesis or activity of Cas9 or level of Cas9 activity influences the repair outcomes. So everybody probably has an idea that the more Cas9 you put into a cell or an organism, the more likely the off-target effects are. And that's sort of readily understandable. Like the more enzyme you have present, the more likely it will work on sites where it's not optimal. So that's not my concern here. What I was wondering if the very concentration of Cas9 influences the outcomes on target, as in the specific outcomes like that. And one way I thought would be one way to tackle this problem was to use this fact that my control clones were a little bit variable, which implies that they had different activities of Cas9, and I could go into details about why, but suffice it to say that there is some variability in control clones, and thus also in the non-hit clones, so to speak, the clones that were knocked out but didn't have any effect. So if you take those and plot them in this PCA-like space, and then the position in this PCA-like space should reflect roughly the indel profile differences. And if you then color those clones in by the efficiency of mutagenesis as measured by sort of direct flow cytometry, a sort of a orthogonal way of measuring that, you would see that the two correlate, implying that the differences in intensity of editing influence the indel profile qualitatively. And to further stress this point, I actually had this nice little system set up with the traditional two-guide or two-part guide RNA, which is much less efficient because of probably the efficiency of putting the two guides together, or two parts of the guide together, versus the sort of standard one guide system that everybody uses. And that allowed me to sort of compare a low efficiency cutting at the same target to high efficiency cutting at the same target. And by sequencing the indel profiles of the two and comparing them, we can see that the strong target is enriched for the largest deletions, whereas it's depleted for the small ones, implying that the more intense the Cas9 action, the more likely the resection outcomes. It's not much of an effect, so compared to the knockdowns or knockouts of key players in non-homologous end joining pathways, it's not a strong effect, but it's still there. And, you think it's an important clue as to how Cas9 itself influences the DNA damage repair. Okay, well, in summary, I hope you are now convinced that large deletions are relatively common and can be very diverse as well. Non-homologous end joining, perhaps as expected, is protecting the cells against large deletions, whereas POL-Q and Nibin, a sort of resection and micromolecule-mediated end joining proteins, are promoting them. And that also means that if you're using inhibitors on non-homologous end joining, that's a good thing to know, that you are running a risk of increasing the frequency of large deletions very strongly. There doesn't seem to be any dedicated large deletion pathway, so I think there is no chance we will have an inhibitor for that specifically. I mean, you, again, could inhibit POL-Q and Nibin, but that probably will have some other effects, maybe some larger genomic instability or cell death. A repair can be very site-specific, which sort of promises ways of tailoring your repair outcomes to what you wanna have, and also in a way that isn't affecting the whole genome. TRX1 is involved in double-stranded repair. We thought that was an interesting thing to find. And yeah, finally, intensive catting does seem to promote resection outcomes, so be careful how much Cas9 you put in. Right, with that, I would like to acknowledge my graduate supervisor, Alan Bradley, and Felicity, who was part of this study as well, on the computational side. All the other people in Sanger and other labs that have helped me throughout this project, and welcome to us for funding this project as well. And please do check out the preprint on bioRxiv, and with that, I'll take questions.
Thank you, Michael. That's very nice work. You call that a small trouble?
Did I? Sorry.
You call these large deletions small trouble?
It was just a very poor attempt at a pun with with small indels sort of being involved, and yeah, I think it's been a big discussion, and I guess we will maybe have it at a panel discussion, right? Like how much do we have to care about on target indels and deletions? But yeah, I guess if someone has this particular questions, I'm happy to.
Right. So I have a quick question. You did most of the work in ESLs. Do you expect the likelihood of having larger deletions to be different in different cell lines?
Yeah, so this is a very sort of fascinating direction that I think I've seen a few labs sort of take in the recent years. So we'll probably learn a lot more about it in the coming years when those papers come out. But yeah, there's definitely a little bit of variability between different tissues and cell types in terms of their DNA damage repair, in particular, obviously the cycling versus non-cycling cells, but then also probably pluripotent cells versus sort of more somatic cells. So yeah, I don't think like everything that we found in embryonic stem cells will directly translate to other cells, but it will translate in as far as most cells use all these repair pathways just to a slightly different degree. So I think it's going to be more of a difference of degree than sort of fully difference of quality.
Okay. Here are two questions. Did you monitor the large deletion frequency over time? Do you think that some of them will be counter-selected?
Well, definitely, and this is something that people have directly observed, and well, for example, in embryos in general, like it's sort of almost a tautology to say that if you have massive chromosomal changes, it's unlikely that would result in a birth of a live organism. So that's been observed repeatedly, both I guess in humans also because people monitor. For example, in vitro sort of fertilization experiments and see that happens. So yes, I haven't specifically been monitoring sort of large deletions in embryonic stem cells. I think they, as long as they are in culture and haven't differentiated, they would tend to maintain sort of potentially weird karyotypes. But as soon as they differentiate, and especially if, say a gene that was important for differentiation is involved, like they would just drop out. So yeah, people have been seeing that, and I think that will be the case to some degree.
The second question is, do you plan to compare the repair outcomes of genes in different chromatin contexts or positions, such as centromere versus telomere regions?
Yeah, so some of the paper that I sort of called out, in particular the one from Culot et al., was looking at cutting relatively near to the telomere, and that was resulting in loss of the entire arm. And you know, again, it's a pretty obvious thing that like you can't lose an arm if it contains essential genes, but the closer you are to the end, the more likely you're not gonna encounter this problem. So yeah, I haven't specifically looked at the regions that are essentially more closed. I mostly looked at the transcriptionally active regions, which also is the type of regions that most people would be targeting. But yeah, there's also a good deal of work on differences in those. In particular, Cas9 can be partially at least inhibited from binding, which would again imply actually that the intensity of cutting, as in perhaps like the repeat cutting and how much Cas9 would be able to bind at any given time is going to be lower, and thus perhaps less mutagenic.
All right, thanks again, Michael.
All right.
Now we move on to our final activity, so our panel discussion. It will be led by Monica Sentmanet from our group and Luke as well. Monica will start the panel with some data in cell lines, kind of echoing Michael's presentation. Monica is an assistant director at the GEIC, senior scientist. And in addition to the mouse work I showed you, she's also in charge of cancer cell line projects. Okay, Monica.
You're muted.
Sorry, I'm muted. I'm going to share my screen here if I can. Let me see here. Okay. Let's see. Not sure why. I'm having some problems. Okay, there it is. Share screen. All right. Okay, we're in business. Sorry about that. All right. So I'm just going to give you a quick overview of our cell line engineering workflow. It comprises much of the work that our core does. We typically have 70 to 80 lines in production at a time. So the operation sort of necessitates a high throughput workflow. Most projects are knock-in, knockouts, and they utilize CRISPR-Cas9, which once we deliver into cells, we validate the pool using next generation sequencing. And this is a two-step process that we use, where we first amplify the guide RNA target site using primers that flank the guide RNA target. These primers have tails that are then bound by the indexing primers. They bring in a unique barcode and the Illumina sequencing adapters that bind to the flow cell, and this gives us some editing frequency. And once we have the validated pool, we single cell sort and genotype these clonal lines. And because we have thousands of unique barcodes, we're able to genotype hundreds of clones for our projects. And this enables us to pick several hits for most projects, which we then expand and perform final QC before we deliver to the investigators. So we've delivered hundreds of projects using this method. And for most projects, we're able to deliver many clones. We've had a few instances when an investigator comes back to us and says, one of our clones is behaving differently. And so what I want to do is show you 3 examples so sort of like real life examples of clones that were behaving differently and what we found when we dug deeper. And okay, and so in this first example, we delivered Cas9 and a single guide RNA by lentivirus. And so the pool is stably integrated and constitutively expressing the reagents for a knockout project. Here we're seeing the total read count with the respective frequencies for each allele. And of these two knockout clones, one of them, C6, was behaving differently. And so what we did was we expanded the genotyping window beyond the DeepSeek primer binding sites. And we identified a large deletion that started where you would expect at that guide RNA target site, but also spanned the DeepSeek reverse primer binding, which is why we didn't detect this allele. And so clone C6 actually has 3 copies. It has the 2 individual indels that we detected, and then now this larger deletion. And so we asked what was the consequence on RNA splicing from this larger deletion. And to do this, we sequenced the cDNA from this clone. And what we identified was it led to alternative splicing. So there's exon skipping here where in clone C6, we have the other isoform or the alternative transcript. We also find it in the pool, but not in the parental line or clone C3. So it's not something that is normally present in this parental pool. It's clearly a product of this deletion event. So in the second example, we delivered Cas9 protein and guide RNAs using it was synthetic guide RNAs. So these reagents are there very transiently. They're gone after 2 to 3 days. And this is for a deletion project. And the strategy was a sequential targeting using paired guide RNAs, where in the first targeting event where we nucleoffect guides 1 and 4 together, we wait a few days, and at day 4, we proceed with nucleoffections with guides 2 and 3. And this effort is to bias the events into biallelic events so that we have homozygous deletion clones ideally. And so you expect when you're screening clones using primers outside of these target sites that you should see sort of 2 distinct size bands. And here are a few of our hits where you see what we would expect is unique products from guides 1 and 4 or 2 and 3. And the other component of our screening is that we're looking for the absence of a couple of PCR reactions that we do within the intervening genomic region that should be absent because we deleted it. The oddity here is clone 3 because we're not seeing this sort of second band that we would have expected from the event from guides 2 and 3. And so we dug a little deeper into that clone. And what we found was that it did have an 8 kb deletion, but that deletion it didn't occur as we would have expected from SP2 or guide 2 and 3. It occurred just from guide 3 cutting, a single cut site. And the consequence of this is that the first exon remains intact in this allele. And so we end up with the same phenomena we had before where exon skipping occurs and we end up with a transcript that's still able to produce protein because this is in frame. And so in this last example, I'm going to show you it's plasmid delivery of Cas9 and a single guide RNA. We also introduced a single-stranded donor to do a knock-in project where we did a C to G single base change. And in this case, we delivered homozygous knock-in clones, but clone A3 was behaving differently functionally. And so we expanded the genotyping window beyond the DeepSeek primer binding sites. And of course, we found this larger deletion, 783 base pair deletion, that spanned the primer binding site from the original genotyping effort. And so clone A3 is actually not homozygous, but heterozygous, where one allele is knocked in and one allele has this larger deletion. And so what I've shown you is, in sort of going with a theme from Michael, that single guide RNAs can definitely induce these large deletions spanning multiple KBs. It's observed across Cas9 CRISPR delivery methods in our hands. Novel transcripts can be generated as a result. And so it's ideal to include multiple hits for downstream functional validation. And that's it. And so from there, I want to sort of segue into the panel discussion and sort of kick that off with Luke. And so that we can hear from the panelists regarding some of the sort of starting with the off-target sort of best practices is sort of the first question we wanted to talk about, which, and in particular, asking Michael, Aaron, for sort of animal models perspective, and Dongsheng, if he's still around, about what your view of best practices for detecting these unintended modifications at target sites.
I'm happy to chime in as from the animal model perspective. These types of large deletion events are not something that we would typically pick up, and so we probably miss them because we're typically just amplifying a small amplicon around the target, looking for small indels. So anything that deleted a primer binding site would be absent in our screen, and we would just count those. Assuming the other allele was wild type, we would just presume it was a wild type founder and would not pursue it further. So at least for the local, deletion, it's not something that we would pick up in a screen. Now, when we do generate models, one of the things that we do take care of is to, very carefully, we try to sequence the cDNA to make sure that there aren't any other kinds of mutations, especially with knock-in models, right? So make sure that we didn't get a sort of offset indel or something, because we have also seen that occasionally where you get an indel that's not adjacent to the cut. And I'm not sure what the mechanism of there, if it's sort of like a failed repair off of the wild type allele that doesn't quite get the homologous recombination to repair a break or what happens there. But especially with the knock-in models is where we do a little bit more work to make sure the gene is intact and all of at the mRNA, cDNA level, we see up the expected splicing pattern.
Michael, what do you think?
Well, one nice thing about like animal models is obviously at some point you breed them, and if you happen to breed a large deletion to homozygosity, you would, well, not be able to detect it anymore, and that's the point where you don't keep this animal anymore, probably. But you mentioned all these, what I like to call non-contiguous lesions or the adjacent lesions that, you would still have, and obviously any off targets you can also breed to homozygosity, and you would never know. So there's a lot of challenges in there. I guess, as you mentioned, like the mRNA sequencing or figuring out the particular phenotype you're interested in and trying to safeguard against it is the best you have. For large deletion, one thing that I generally want to mention is they do, and no one has really done it systematically, but from the little data that I had, it looked like it's very, like it decays exponentially, the frequency of deletions with their size, right? So the larger ones are exponentially less likely than the small ones. And that means that, if you say are able to do a 6 kb PCR just to see if anything looks wrong, you've probably captured like most of possible large deletion outcomes at the very least. So even that, and then another option is obviously copy number qPCR. If you can't do the whole karyotype, which would sometimes tell you if there's some chromosomal duplication, you can always do the copy number qPCR around the cut site, and that would tell you something's wrong.
Right. Right. So just to follow up, Michael, do you have a sense of what you think is sort of the biggest determinant of the frequency of these outcomes? Do you think it's like locus specific and maybe there's some sequence determinants there? Guide RNA, I guess we kind of talked a little bit about cell type and maybe not knowing necessarily how much of an influence that is. What's sort of your take on sort of all the possible inputs?
Right. Well, obviously, yeah, it does seem definitely that DNA damage repair is involved, as in if you have a cell that is non-homologous end joining sort of deficient, then you would expect more outcomes like that. And again, it's not a very strict science yet, but it seems like, say, pluripotent stem cells are more like that. Even though they can do non-homologous end joining, they're not super proficient at it. With respect to the guides in particular, I mean, I can only speculate that, that there does seem to be guides that tend to end up with homologous end joining, with non-homologous end joining versus microhomology mediated end joining, and that's been sort of a subject of a flurry of paper, partially from Leo Parts from our institute, but also a number of other papers that came out at the same time trying to predict the Indo profiles. Whether that correlates with large lesion frequency is an open question, but it's an obvious sort of direction to take and ask that I would do if I had my own lab, but I don't. So, yeah.
Okay. Right. So I'm going to sort of switch over to let's see, sort of disease models. And so just going into some of the questions for Andrew Yu and Martin about sort of iPSC differentiation and versus directed differentiation from primary cells. Can you talk, I guess, specifically, Andrew, about general guidelines of sort of which to use, what cell types, what types of diseases to model?
Yeah, I think so. In terms of taking examples from model Huntington's disease, what we learned was that coming from the same patient samples, if we turn cells into different types of neurons, for example, if we turn towards cortical neurons versus if we compare them to the same fibroblasts now turned into medium spiny neurons, and if we measure the neurodegeneration and the level of cell death and whatnot, they're radically different, meaning that it's actually quite important to have a control for what kind of neuronal subtypes that you can generate. So it's the direct conversion, I think is a good tool for modeling that kind of sort of late onset aspect of diseases. But one caveat, and it's a part of the experimental step that I really envy the ability to be able to expand the cells, because what happens is as soon as, so starting from the fibroblasts, as soon as the direct conversion is induced, the first thing that happens is cells come out of cell cycle. So that really limits two applications in terms of using this for using these cells for either any degree of genome editing approaches, because we are virtually looking at like almost all cells being post-mitotic, and using those cells for any type of cell transplantation experiment, because we're very limited in terms of how many cells we can generate. So the application of these cells is more limited in terms of what you can do with them. But one huge benefit is the ability to be able to model sort of the late onset of the disease.
Do you see a difference when you're, I mean, because you're looking at age differences. You have fibroblasts, I guess, from young, old patients, people that are affected. Like, do you see a lot of variation in your ability to expand these different flavors of fibroblast lines?
Yeah, so neonatal, in terms of the cell proliferation index, neonatal cells, they divide a lot faster. So, you know, so for some project going on in the lab where we try to knock in reporters specifically into, you know, some neuronal markers and whatnot, we tend to use neonatal cells because they're much easier to proliferate, but they still have you know, limited passage numbers that they can go through. There are the tricks that can bypass that, so we can sort of semi-mortalize the cells with hTERT. Then you can sort of use them as a somewhat like cell line like approaches, but adult cells are more tricky. We cannot go over, let's say, passage 20. We're very limited. So actually, my lab may be like blacklisted in terms of requesting the same line over and over from NIH. Yeah.
Right. Got it. I think we're running a little bit over on time, but let's see. I have one more question. So what are your, for Andrew and Martin, do you feel that there's like a complementary role that iPSC derived differentiation versus directed differentiation from primary cells can play in modeling age-related disease?
Yeah, maybe I can take this one. I agree with all the great points that Andrew made. I think the direct converted cells are a very attractive model. And I think as with all models, you know, there's often a trade-off between tractability, scalability on one end, and maybe physiological faithfulness on the other hand, right? So as Andrew said, I think one of the strengths of iPSC-derived systems is the expandability, so that we can really do genome-wide screens in highly reproducible cells. We can engineer them to make isogenic controls for disease variants, and those are very attractive features. And so I see those as great models for the kind of systematic, massively parallel screens that we often like to start with. But then I think it's, for any kind of model system, it's really important to validate your results in maybe even more physiological model systems. So I could see that at that point, you know, looking at direct converted cells, and of course other models too, right? Validating some things in primary tissues or animal models can all be powerful ways to establish that, you know, results from your tractable model have broader applicability.
Yeah, I totally agree. And also the compatibility between different systems may offer insights into what goes on, the younger aspect of, you know, lifespan and older, and we can have this kind of comparative analysis between two systems and figure out what's there in one system versus not, so that we can try to sort of mimic. So for us, we are trying to go younger from old cells. So we actually try to learn from iPSCs and neurons, and we try to mimic those neurons by going younger from old cells.
Can I have a quick small question for Duke? So I know, like, you know, fibroblasts and iPSC, they are both really good model. So it's possible, like, use those, you know, you can control the methylation with the Cas9 approach, try to maybe turn back the clock to manipulate those epigenetic clock for, you know, from iPSC and to, you know, to reflect their age signature.
Yeah, I mean, I think it's definitely possible. So I'm not an expert on the clock literature, but I don't know that there's a thought that the clock, like the methylation sites that define the clock, I don't know if those are considered to be causal for aging phenotypes or if they're just correlated. And so if they're just correlated, they serve as a great clock, but reversing them wouldn't do anything. I also am not, I know that originally there were something like a couple hundred sites that defined the clock, and I don't know how few they've gotten down to. So maybe Andrew can also answer this question. We have not targeted more than 4 protein coding genes with CRISPR-off, although we have used the dCas12 CRISPR-off successfully. And so there's recent work from like Randall Platt's lab that has used Cas12a to mutate up to 25 sites. So in theory, you could target like 20 or 30 sites. I don't know if you had to target a couple hundred sites, that would, I'm not aware of anybody who's targeted a couple hundred sites with Cas9 or dCas9 applications. I think the most I know of is the Church lab mutated all the pig endogenous retroviral elements, and there were like 60 of them, but yeah, nothing beyond that.
Yeah, I think it's with kind of extending Luke's point. I think right now it's safer to assume the clock is more correlation than causal, and it's hard to answer that question mainly because we don't know what molecular machinery is responsible for causing these differences in DNA methylation patterns. So once we find that what's responsible for it, and if we can perturb, and then if that actually rejuvenates the cells physiologically, then I think we can assume that the clock is actually responsible for age-associated phenotypes. But until then, I think it's safer to assume the clock loci to be more of a marker than causal for aging.
If I may quickly interject, Wolf Reich had a bioarchive paper just a few days ago where they claimed that like reprogramming, I think it was fibroblasts back to iPS cells and then back to fibroblasts, like resets the clock, which was interesting because in your case, I think that wasn't the case, or at least sort of a similar. So anyway, I'm just posting the link. I have no expertise to really tell how good this experiment is, but maybe it's relevant.
Yeah, phenotype wise, we actually did an interesting experiment where we start with adult fibroblasts and turn them into neurons, and we detect some phenotypes. And then we also did the same thing by actually generating iPSC first, but redriving fibroblasts out of it, and then use those fibroblasts, more kind of younger fibroblasts, and do reprogramming and compare basically two population of neurons directly coming from adult side versus young side, and the phenotype changes a bit. So phenotypically, I know the fibroblasts that were derived from iPSCs that came from adult cells, they're actually quite different. But the clock, yeah, clock, I don't know. We didn't measure that.
So the other thing I was going to sort of switch back to is sort of the idea of alternatives to knockout, to NHEJ based knockouts, and asking Luke and Alejandro or Alex for their take on sort of replacing, or how close we are to replacing NHEJ based knockouts using, you know, sort of the kind of mechanism that Luke presented on, so that we can avoid these sort of larger deletions and issues that we encounter, even in screens like you guys showed with P53 being an issue.
I can go first, and Luke, I'm sure, can follow up. So I think when CRISPR-I first came out, it was pretty clear that it probably wasn't as powerful as Cas9. Some of the original papers kind of compared them head to head and it just didn't look as good. I think we've made a lot of progress. I mean, Luke's story is incredible, right? I think if that really holds up across all these different cell lines, it could change the game, because Cas9 is amazing, but the problem is the indels can still be in frame. You get exon skipping. you've got all these kind of weird ways you can kind of escape the selection. And I think CRISPR is going to really help solve a lot of those problems. Also, long non-coding RNAs, I think it really opens up that whole area for exploration much easier. And then again, I think, dose titration, I think it's just a more programmable tool once it becomes robust. So I do think there's actually a future for it, especially if you're gonna do like combinatorics. Cells don't like 5, 10 different cuts in their genome at the same time, and they're gonna get really angry. But I think again, repression seems to be quite a multiplexable tool. People, I think, Jay Shinder had a paper where he did like 20 some different repressions at the same time in cells, and it looks like they didn't explode. So I think the tool has quite an interesting future. Luke, what are thoughts you have?
Yeah, so I have a lot of thoughts and a lot of opinions, not all of which are shared by the community. I think Cas9 is easy, right? As long as it's nuclear and you express it long enough, you'll make indels and that'll knock a gene out. DCas9, on the other hand, I think of more like GFP, where 30 years after GFP, we continue to derive new fluorescent protein variants. And I think DCas9 will be the same way as either an epigenetic editor or a programmable transcription vector. It's just, there's so many ways you could use this to control transcription or genome topology or epigenetic code or whatever. So my guess is there's going to be a lot more to do. And I know Alejandro's working in this space, or there's a lot of exciting options here. The second thing is related to the early comparisons between Cas9 and CRISPR-I. Some of those, and I'll call this out on purpose because I still am a little bitter about it, from René Bernard's are really poorly done. So they intentionally used poorly active sgRNAs for the CRISPR-I arm, and the wrong sgRNA scaffold and the wrong algorithm. And so this has been done repeatedly, where if you want to break CRISPR-I and use a bad fusion protein that isn't an active epigenetic editor, or you want to use hypoactive sgRNAs, you can set up like false arguments basically that claim that something is better. This is true also for some of the recent CRISPR-I work from the Taipei lab, where they claim that a ZIM3 fusion protein is more active than a COX-1 fusion protein. Wow. If you look in their supplemental data buried in one of the supplemental figures, as soon as they switch COX-1 or ZIM3 to the C terminus, they're equivalent. They're like within 1% knockdown of each other. So they set up this major paper around something that their own data doesn't support, because all their N-terminal fusion proteins are sort of crappy epigenetic editors. So I don't think that's really very fair. And I don't think most people read every last supplemental figure. And so the lesson is the devil's in the details with this. And if you can publish a paper, but like, do you really want to be known for that? So that's pretty opinionated. I'm being way overly honest here, but there are better CRISPR-I fusion proteins and there are worse CRISPR-I fusion proteins. And so be careful what you're using.
Luke, do you see CRISPR-off going to therapy one day?
Yeah, so that was part of my disclosure slide. I do.
Okay.
Okay. I think.
Can I also ask a question too? So I was wondering whether if Luke sort of tried to compare between DNMT3A versus DNMT3B, any functional difference between those two?
No, we should have done that. There's a lot of literature on differences for A versus B. We're actively looking into this in the context of an atypical form of methylation that's been suggested to be deposited by A versus B in iPS cells or ES cells. We should have done this, honestly, yeah. Great. I don't have any data for it. It's a really good point, though.
Okay, so I'm going to start sort of the last sort of question is for Aaron, and just sort of asking about plans to sort of put these models in more hands. You know, what kind of sort of efforts your group has to do that?
Yeah, thanks. Well, I think it's a big challenge not only for our group, for any anybody who's exploring CRISPR in a new model system, right? The infrastructure that the scientific community has for warehousing, distributing, advertising, commercially, selling genetically modified mice is large, and it's there's huge efforts and a lot of people involved. I think what we can do is try to, one thing that we're really focused on is trying to build sort of like groups of models to try to attract a group of, a field of investigators. So for instance, we just built a group of autism models. Some of these, Sage had produced in a Sprague Dolly background. We were getting a lot of requests to have them in a Long Evans background. So we kind of recreated some of those models and have like a resource. And we're starting to see like attraction in communities of researchers who are interested in having those as well. And so that's one way is, and I think we could do a better job of marketing those little niches. Other than that, I mean, we do have, the RRC in Missouri where we can deposit strains. I've been talking with Elizabeth Breida on and off about, we need like a central one-stop shop for rat models because there's, and we could involve commercial sources as well because I think it's too scattered right now. And one of the big concerns that I have is the duplication of models and in research, and not only with rats, but also with mice and other species, right? It's too easy now to create a knockout mouse. You don't need to go see if there's one out there already. You can just have it generated down the hall for quite cheap and have your very own mouse. And I guess, one of the things that's concerning about that, right, is the amount of resources that need to go into characterizing a model, and how, and NIH is catching on to this. They are catching on to this. And are they going to fund you to keep making new mouse models that already exist? Maybe made by ES Technology something they've already invested in years ago, right? So I think this is something we have to pay attention to as a community. In the rat world, as I mentioned, it's a little bit different because many of us, or most of us, are not working in the same strain backgrounds, whereas maybe in the mouse, it's much more shifted towards C57 Black 6, and a lot of the strains that you get are in that background. So we have the issue of, well, I want the gene knockout in SS, I also want it in the SHR, and this other lab wants it in this other strain, right? And instead of breeding the mutation from one strain into another, we tend to just start over and create a new model. And one of the concerns I have, again, is how much duplication is happening in the community. How do we get our models out there quickly as soon as we can make them available for sharing, and get them out in a place where people can find them, so that we don't create this problem that down the line that we can't support, we can't possibly maintain them all. So those are my thoughts about animal model resources.
If I may have a little plug in here, I just want to sort of generally bemoan the fact that especially UK has really gone back on like animal models, especially mouse models. So both Wellcome Sanger Institute and the MRC Harwell are essentially out of business, which I find like extremely contradictory to everything I hear from, say, ENCON consortia and places like that, which have 10 bold predictions for genomics, and one of them is we need to understand what genes do. And like, how are you going to do that if you're like literally turning down like the most valuable resource for understanding them? Yeah, sure, you can understand them in cell lines, and it's super important, and again, work like Andres or Martin's, like great work, very important. But there's going to be there's bound to be other phenotypes, right, in the whole body when you knock something down or modify it, and you can't know it without an organism.
Mm-hmm. Yeah, that is very been very disappointing. The rat community in particular, people who are study rats and want to generate rat models in Europe have basically vanished. We create some and we ship them overseas. I'm sure others do as well. But yeah, those they're not getting the support they need there.
I'm hoping, Aaron, after this, because I get the sense that around here, really not that many people knew there are so many models in rats available already. After I came here, that's almost 4 years now, we made 2. Because people are just not really in the mindset to even think rat models is a possibility. So I'm really hoping you will get some emails.
I hope so too. I mean, you know, one thing that we've learned along the way is, right, it's one thing to create a model and put it out there. If there's no published phenotype, right, people are pretty gun shy about it, right? They want to see a phenotype. They want to know if we've proven this or that about the model. And, you know, to be honest, because we make so many models for others, you know, we have a bank, we have a large resource, a large bank of these models, but we haven't phenotyped them all. That takes years, right? And teams of scientists. So that's another part that we run into is, you know, what phenotypes have you observed in this model? And it's like, well, we created it as part of this resource program. You know, you'd have to go ask the original investigator who requested it if they've characterized it, if they're getting ready to publish it. And you know, with rats, it's slower than mice. It takes longer to breed. But I think it's just the ability to create these models has so far outpaced the ability to characterize them and get them out there with known phenotypes. And so that's a challenge.
Cool. Anything else, Monica, you want to cover?
I think that covers the topics we wanted to chat about. Yeah.
Cool. I think we are done. I want to thank everyone for taking the time to participate, especially all the speakers, for sharing all the impressive data and great discussion. I really learned a lot from you. I hope many of you feel the same. The talks and posters will be on our website. I think Candice will send the links. I really hope next time we have this meeting, we will be able to meet in person. And I wish everyone a very productive new year.
Thanks so much.
Thank you, guys.
Thank you. Bye. Appreciate it.
You guys are out.
Okay.
Bye.
Bye.